Hello, and thank you for taking the time to review this recording of the 2025 Invasive Shothole Borers Fusarium Dieback Symposium. I'm Randall Oliver, California's statewide Invasive Shothole Borers Communications Coordinator and a member of the UC Integrated Pest Management Program team. My role in this meeting was initially to introduce our illustrious presenters and to help manage things on the back end. And given that we were trying to do a hybrid meeting in a remote location, we anticipated that we would run into some technical issues, as this slide clearly indicates. However, we did not foresee the magnitude of those issues, which included a failure of most of our first presentation to properly record. Therefore, whereas people who attended the September 23rd presentation, either through Zoom or in person, had the opportunity to hear from Dr. John Kawashima, a University of California Cooperative Extension Environmental Horticultural Advisor, Emeritus, and an icon in the field of the management of insects, diseases, and weeds. You instead will have me to quickly review most of John's presentation with a brief cameo from John at the end. During our attempt to resolve some of those issues during the meeting, John quipped that I might be responsible, as I would do just about anything to get more attention. I assure you that that was not the case, and I hope that you will bear with me for the next few minutes. I'm sure I will not be nearly as entertaining as John was. John's comments focused on the biology of the beetles and the fungal complex. The term invasive shothole borers is a common name that refers to several species of ambrosia beetles that are native to Southeast Asia. These beetles are very small, about the size of a sesame seed or Lincoln's nose in the photo. For the past 2 decades, we have focused our attention on 2 species that were known to be established in California, the Polyphus shothole borer and the Kuroshio shothole borer. Morphologically, these 2 are nearly identical, and we need DNA identification to confirm the species. Now we also have another species that until 2023 was unknown here, the greater shothole borer, which is larger than either of those 2 species. Dr. Shannon Lynch discussed that in detail later, and John's comments focused on the first 2. The term ambrosia beetles means that their food source is not wood, but rather symbiotic fungi that they bring with them when they tunnel into their host trees. That fungus is the main thing that kills the tree host over time. This is why we refer to this tree-killing combination as invasive shothole borers Fusarium Dieback complex. Fusarium refers to the main type of symbiotic fungi, although there are also others. Unlike the majority of ambrosia species, these beetles attack apparently healthy trees. The females make galleries in the wood and they lay their eggs. They inoculate the fungus into the gallery wall at the same time. Now, the female decides the sex ratio. She typically produces far more females than males within the galleries. If she lays unfertilized eggs, haploid, those can turn into males when hatched, and they hold on to the sperm and fertilize as they lay their eggs and choose the number of female eggs that they wish to produce. Brothers mate with sisters, and this eliminates the risk of needing to look at the opposite sex outside of the tree. And for this reason, this class of ambrosia beetle does not produce sex pheromones, which has important management implications. They prefer to produce a new gallery on the same tree near their mother's gallery. The fungus, meanwhile, continues colonizing the wood beyond the gallery wall, and the fungus can cause branch dieback and tree mortality. When the new host can no longer support the growth of the fungus, the adults leave that host to find a new host, and they start the process all over again. Now, like other invasive species, shothole borers have the ability to rapidly multiply. Assuming that just 5 females survive from each brood, you could have close to 100,000 beetles in that tree in just 7 generations. It takes somewhere between 25 and 50 days for the larvae to grow into pupae and become adults under ideal temperature conditions. Models based on the effect of temperature in their development suggest that these beetles can have 5 to 11 generations per year in Southern California. So this can happen very quickly. So which kind of trees are susceptible? There are 247 species of trees that are attacked, 78 species of trees that can support ISHB reproduction, and 17 trees that are highly susceptible and can be killed. You can see this chart as well as more information in the UC IPM Pest Note, which can be found online at ishb.org under resources. So what is the potential impact of ISHB FD? In severely infested trees, the beetles and their associated fungal disease can cause tree decline, branch dieback, and tree death. Trees with heavily infested branches can be especially hazardous since the combined damage to the fungal disease and the beetle's tunneling activity weakens the wood, causing limbs to break and fall. And dead or dying trees can also provide fuel for wildfires. It's a big issue in California. A 2017 study found that a third of Southern California's 71 million urban trees are susceptible to ISHB FD, and if just half of those die, the replacement cost would be nearly $16 billion. That doesn't include lost ecosystem services or lost aesthetic value, which were looked at on a smaller scale analysis done a couple of years ago, and you can find the details to that on ishb.org. And also, those numbers were limited to Southern California. And as you may have heard, the beetles have now spread to the northern half of our state. So the numbers are very large and impactful. To review, as the fungus spreads, it leads to branch dieback, and it can spread very quickly in some species like box elder, which you see here on the left. As for the trees become severely infested, what we call amplifier trees, they become safety hazards and a source for beetles to infest other trees. And we've seen devastating consequences of this in the past in Southern California, in the Tijuana River Valley, where we saw 140,000 willow trees severely damaged, and in Laguna Niguel in Orange County. There have been other dramatic infestations, such as at UC Irvine, where nearly 1,000 trees were removed, and many of the now available treatment protocols were developed. So visual surveys are critical to stopping the spread and managing this pest. But the beetle is small, and you probably aren't going to find it unless you're looking very closely at a severely infested tree, which is something that the group did in the afternoon following the symposium. So if you are out looking, what should you look for? The term shothole borer comes from the distinctive entry holes that are perfectly round and about the size of the tip of a medium ballpoint pen. Other signs and symptoms include staining, boring sawdust or frass, gumming, and sugar-like buildups. Signs and symptoms can vary with the species of the tree, and you can learn more about these on the website. But it's important to remember that even if you do see some of these symptoms, it isn't ISHB unless it has the distinctive entry holes.
If you look at avocado, it'll start to push out a lot of fluid, and then it's mostly kind of alcohol that flashes off and leaves behind sugars. We call that the sugar volcano. And something like brachychiton that really saps will sap out the beetle. Brachychiton are actually fairly resistant to this beetle. But the problem is, it's not always one thing that causes a tree to die. It's oftentimes multiple things. It can be a cascade of events, simultaneous. But what happens with the brachychiton at UCI, they had a row of brachychiton all on the same irrigation line. One tree got hit heavily with shothole borer attacks, and it fluxed out so much gum that that tree was weakened. And so now, all of a sudden, it didn't need as much water as the other trees. And we think that it actually was so wet, it died because of overwatering and possibly Phytophthora root rot. So on sycamore, if you shave off the bark, then you'll see this, on a healthy tree, you'll see green. If you shave off the bark around the hole right there, you'll see that there's fungus in the gallery, and then the fungus is moving out. So we oftentimes will shave off just a little bit and look to see if we find this. If you do, and you want to confirm it, you can either try to find the beetle and send that in for analysis, or you can actually take the sample of this infested wood and send it to a laboratory, and they can identify the fungus, because Fusarium is, remember, closely related to the beetle. So we can often tell if it's Corosium or if it's the Fusarium dieback by the pathology lab analysis, and that's what you would do. So when the trees are heavily infested and they start showing dieback, these are now considered amplifier trees. And amplifier trees need to be removed, because if you don't remove them, they're going to just be pumping out thousands of beetles into the area, and those beetles will keep trying to find a suitable host. So we have a committee which was funded by the Assembly bill they talked about, and we received $5 million, and it codified 2 committees, the California Invasive Species Advisory Committee. So this is the working committee to the Invasive Species Committee. The Invasive Species Committee consists of all the secretaries of all the agencies in the state of California. So they do the work, and then they go to the Invasive Species Committee and they give the blessing. Yes, we'll fund that. So we received $5 million to codify these 2 committees. And then we had a real thorough process of determining what type of research was needed, because there was no funding initially when we started working on this. We were getting money here and there and dribs and drabs. Avocado Commission, I have to give them a lot of credit for the money that they supplied to get some of our initial data. And then once we received that funding, we're able to come up with more research projects and questions than we had money for. So that's why we're continuously trying to get money. We came up with some research money for early detection and rapid response. We were looking at things like pathways. This beetle is very tiny, about the size of a sesame seed. There's no way it flew like polyphagus and Corosio beetles are down here in the south. When we find these beetles hundreds of miles away from the initial infestations, that means it didn't fly. Pathways, it's human-mediated transport. People move this beetle around in green waste and in wood. So I have another photo of a guy in a Mercedes convertible with firewood stacked up in the back that he's moving from somewhere. And there are a lot of agencies now who have become involved in this. So we have the Department of Food and Agriculture, UC Cooperative Extension, which is the outreach arm of the University of California, Cal Recycle, USDA, Forest Service, Cal Fire, and then blocking that one back there is a very unimportant agency. What agency was that? I don't know. Why is it blocked? So here's the key takeaways. It's not going away. It can't be eradicated, but it can be managed. And Keith and I implemented a shothole borer IPM program at Disneyland, and we have kept that thing managed for many years. And it shows up, and it's like whack-a-mole. Shows up, it comes down. Doing nothing is not an option. Before the pandemic, Disneyland set up a trap line in the Anaheim parks surrounding Disneyland. We felt that maybe they might reinvest us. Before the pandemic, the trap line was up. We're monitoring this one specific park that's barely infested. During the pandemic, we cut the funding for that project. And after the pandemic, when we came back, they were removing amplifier trees. So it can happen that fast. If this thing has, what's the state called, 9 to 20 generations per year?
5 to 6.
5 to 6?
In Southern California.
That's not. No, I don't like that. 9 to 20. So if this can have 5 to 6 generations per year, and you saw that graph, in 2 years, this can build up to high enough numbers to take out a tree. That's what happened. If I'm getting the cutoff, I don't care. I'm up here on the boss. So you need to do continuous monitoring, because we're having this meeting here because they found it several years back. They treated it, they walked away, nobody looked for it. You're going to see infestations at Cobb Sports Park like you've never seen before. And it is like whack-a-mole. It was in Laguna Niguel for a while. And then it went to Anaheim, and then it went to Irvine. It just moves around. And then I had a lot of slicers that I know Randall took out. But the one I want to emphasize is the Tijuana River. And are you going to talk about that? And my prompter's not talking about it. So Tijuana River in 2015, 140,000 willows just hammered. And we were very concerned because that's habitat for endangered species. The thing about willows is they can resprout from the base. And so in a few years, it resprouted, and there was enough vegetation there that at least burial were able to reproduce. It happened again very recently. Shannon showed me a photograph, and I went to Google Earth and looked on a map of that area, and it showed that it got hit again 7 years later. And you can see where it turns all brown in that river, and you can see the next year it's all green, but what you're looking at is resprouting. So over there, it looks like it might be approximately a 7-year cycle where you can think it's gone, but if you don't keep looking, it's going to build up to where it can knock everything out again. 140,000 willows are probably infested again. If you look at Google Earth, it's all brown. And then you look a year later, it's all green. But if you ground truth it, that's all we sprouting. So I'm finished. Thank you very much for your patience. And I guess you're ready for your next.
So our next speaker is Dr. Shannon Lynch. Shannon is an assistant professor at UC Davis's Department of Plant Pathology. She's a past chair of the California Invasive Species Advisory Committee and is currently co-chair of its Invasive Species Borers subcommittee, along with Dr. Berman, who's also one of our presenters today. Shannon's worked extensively in California, studying invasive shothole borers and developing a statewide strategic initiative to control and manage this pest disease complex. And she even continued her ISHB involvement while she was serving across the country as assistant professor in forest pathology at the State University of New York College of Environmental Science and Forestry, and that's a mouthful. She was the 2023 recipient of the Western chapter of the International Society of Arboriculture's Award for Arboriculture Research. So, Shannon?
Good morning, everybody. Thanks for being here today, and thanks for the opportunity to talk with you about a new beast in town. I'm going to present on this Eualacea interjectus that carries another fungus called Fusarium floridanum. So let's get into it. So John talked about the polyphagus and the Borers shothole borer, and I'm going to talk about what we're referring to as the greater shothole borer. So this is a new detection that occurred in Santa Cruz. And just to give you a little bit of a background, John did a good job of giving you an overview and alluded to this infestation that occurred in Northern California of the polyphagus shothole borer, which was confirmed in November of 2023. So this was the first detection of the polyphagus shothole borer 280 miles north of the leading edge of the infestation, since it had spread within Southern California. And then in October of 2024, a separate infestation was discovered in Felton, which is just north of Santa Cruz, on box elder trees. And so this was thought to have been the another detection of Polyphagus shothole borer. But in November of 2024, CDFA labs confirmed that this was a new introduction of a third shothole borer beetle pathogen infestation complex, which we're calling the greater shothole borer, that carries another Fusarium species called Fusarium floridannum. So this beetle is much larger than Polyphagus shothole borer. So it's 3.78 millimeters long, in contrast to the Polyphagus shothole borer, which is 2.1 millimeters long on average. So these are the beetles on a penny. So here's greater shothole borer on the left and Polyphagus shothole borer on the right. And you can see there's a real major difference between the two when you look at them side by side. The entry holes are a lot bigger as well. So we're talking 1.7 millimeters in diameter versus 0.83 millimeters in diameter. So for reference, if you're taking the your pen, your ballpoint pen, what we usually say with the Polyphagus and Crocio shothole borer, the tip of your ballpoint pen is the size of an entry hole. Well, you could put the whole ballpoint pen into the entry hole, and that would be greater shothole borer. So that is how you can tell the difference between the two. Fusarium floridannum, this is the what if we culture the fungus from the mycangia, these are the colonies that get produced. This is the fungus grown in culture. When we basically, so we talked about how they carry the fungus from tree to tree. They carry those fungi in special mouth parts called mycangia. So what we do is we cut off the heads, and it's very civilized. We cut off the heads and we plate them out onto culture media to grow, to see what kind of fungus they're growing. And that's what the fungus looks like in culture media. And then these are what the spores look like. They look like dolphins. Thank you, John. Oh, I do?
Yeah.
Oh, look at that technology. Okay, so so they look like little dolphins. So this is a specialized characteristics of the kinds of Fusarium fungi that the Eualacia beetles carry. In addition to the Fusarium floridannum, these beetles also carry another species called Paraacromonium. Polyphagus shothole borer also carries Paraacromonium. We don't know what the role is of these fungi, but it's interesting that they consistently are carrying these other fungi in the mycangia as well. So this is a map that's showing all of the Eualacia species. So Eualacia is a complex of species, ambrosia beetle species that include Polyphagus, Crocio, and a whole bunch of other beetle species that originated from Southeast Asia. This map is showing where they've been introduced from all over the world, including Polyphagus and Crocio shothole borer. Not all of them attack living and healthy trees like Crocio and Polyphagus shothole borer, as well as the greater shothole borer. But they all are they all have this special symbiotic relationship with these Ambrosia fusarium species that have those dolphin-shaped spores, and that is very key. So the greater shothole borer was introduced to the U.S. from Southeast Asia on 3 separate occasions: in Hawaii in 1976, Louisiana in 1984, and Texas in 2011. So it was basically spreading throughout the Southeast United States, established throughout the 80s. So this detection in Santa Cruz is the first occurrence in the Western United States. So this is our first case that we've had of this beetle in the Western United States. It's been found to be detrimental also to populace in Argentina and China, as well as Japanese fig in Japan. So we know that it affects box elder in the southeastern United States as well, but we don't really know much else about its host range. So here is where we found it. It was found originally in Santa Cruz County. So originally, so this is the map of Santa Cruz. This here is in the Felton area. This is Henry Cowell Redwood State Park. This is the location up here, and here we are zoomed in. This is a wood processing area, and this is the original detection. So this location here, the Zion Trail, is where the infestation spread, primarily running through box elder trees. And so the infestation spread around, like throughout this whole riparian area through box elder and into Henry Cowell Redwood State Park. And then in September 4th of this year, it was found 4 miles down the leading edge of the infestation, which is important to understand. This is the mouth of the San Lorenzo River right here. And this river is basically a corridor that moves all the way into Santa Cruz, the city of Santa Cruz. And so this finding here is just a mile into Santa Cruz, the city of Santa Cruz itself. And then in September 10th, it was found in an area just at the north end of Santa Cruz, the city of Santa Cruz. So it's officially established in Santa Cruz as of September 10th. So it's moving pretty quickly within a year. So like I said, it's moving mostly through box elder. These symptoms look exactly like you would see with Polyphagus shothole borer, Crocio shothole borer, the staining that surrounds a entry hole, but the entry hole is much larger. So it looks very similar. It's moving through box elder trees, but we still are looking at the broader host range. This is looking at box elder in cross section. And again, you can see the galleries are going to be much larger as well than one would see on something that's caused by Polyphagus or Crocio shothole borer. So this is the gallery that's being produced, and then you see the fungal necrosis, and then the staining that's being caused by the Fusarium as well, that pigmentation that's caused by the Fusarium that's colonizing the wood. And here, we're looking at this in cross section, but these are coming from galleries at different points along the tree. But these galleries serve essentially as entry holes for other canker pathogens. Once the galleries are abandoned and everything like that, they can serve as entry holes for other canker pathogens to come in and cause further damage to the tree. So here, there's already some canker pathogen that's coming in and colonizing the tree here and producing even more damage. So there's these secondary effects that happen as well. This is a box elder that is showing, this is about a 60 centimeter diameter tree that is at the site along the Zion Trail. It's riddled with entry holes. There's thousands of entry holes on this tree. It is clearly dead, and this is just an indication that the beetle has been established at this site for some time now. So in response to this infestation, Santa Cruz County Department of Agriculture and UC Cooperative Extension are coordinating with CDFA to monitor and respond to the infestation. And then my lab just received funding from CAL FIRE, and we are collaborating with Cooperative Extension and State and County Parks to use this funding to establish permanent monitoring plots to basically track this infestation and understand the disease dynamics over time, as well as optimize monitoring traps. This is important because we want to, the quercevorol traps, the quercevorol lure is what is used to trap Polyphegus and Crozier shothole borers. But because this beetle is larger, it might respond to different concentrations of quercevorol or other compounds as well. And so we're going to do the work to optimize those traps so we can monitor the beetle effectively throughout the region. And we also need to determine the actual host range of this beetle as well. Important reason for that is because it is primarily running through box elder, but one thing that we know, the lesson that we've learned with Polyphegus and Crozier shothole borers is that we know that they have a very broad host range, but when the Polyphegus shothole borer, we'll just use that as an example, establishes at a site, oftentimes it will pick one or two hosts to establish in that location. And it will prefer that host and run through it. But once it's kind of run its course through that particular host, it will jump to another host. Early on in the infestation with Polyphegus shothole borer, when we were establishing its host range, we had heated debates about whether sycamore was a reproductive host. And now we know that sycamore is like driving the epidemic. And so that's just kind of a lesson that's learned from this case. And so right now we have established pathogenicity tests on the box elder on site because we need to just, we are confirming that the fungus is causing the disease, just to kind of cover our bases there. But we are also seeing attack on other tree species. We're seeing it on red willow and coast live oak. We're seeing it on Pacific willow as well. And we are seeing these probing galleries. When you cut into the tree in cross section, these probing galleries. So it is establishing on these trees as well. And we're seeing it on black cottonwood as well. So what's interesting with the black cottonwood is we're not seeing the staining symptoms that you typically see with, Polyphegus and Crozier shothole borer, and that we would see on a sycamore, for example. But we see like lots of entry holes. And when you peel the outer bark, you see that necrosis on the phloem that's associated with these large entry holes. So we are seeing other tree species being attacked, and we want to establish the full host range. So we're going to be doing no choice tests to establish host range on like 50 tree species, and we're going to be doing that experimentally. We're also seeing attack on western sycamore. These are just, again, a few individuals, one hit here, one hit there, but we want to be cognizant that this is happening. We are very early in the infestation here, but that's where we're at. Graphimulalacea is another fungus that Polyphegus shothole borer carries in its mycangia. And it is not found, I should say this, the predominant fungus is Fusarium Dieback. Okay, so Polyphegus carries Fusarium Dieback, Paraacromonium cambium, and Graphium uolacea. Those are the three fungi that Polyphagous shothole borer carries. Okay. The dominant fungus is Fusarium uolacea, but the Graphium is also important. We know that it is somehow supports the larval stage of the reproductive life cycle of the beetle. And so sometimes you're going to recover that fungus because it's an important part of the life cycle.
It's interesting that the CDFA has rated it. It's rated differently than the Fusarium.
Yeah, it's a weak pathogen, but just like Fusarium is a little like colonizes better, but the Fusarium is the dominant species in the mycelium. We've done the studies where we've counted the colonies and things like that that grow in the mycangia, and the Fusarium is dominant in the mycangia. And so it's the primary source of food that's supporting the life cycle. It really relies on the Fusarium to grow and reproduce.
Shannon?
Yeah.
Hi, it's Julie. I'm reviewing questions as they come in. Just a reminder for the people online, please put your questions into the Q&A. And the question is there potential for greater shothole borer to move to Southern California? And if so, what would that look like beyond the impacts from invasive shothole borers?
That's a great question. So the potential is there, just like the potential for Polyphagous and Kuroshio shothole borer to move to Northern California. Like we've found Polyphagous shothole borer in Northern California. The impacts are contingent on what the host range is. And so that's something that we need to determine. Okay, so now we are moving on to the talk about the current distribution and threat map. Okay, so we're going to talk about the current distribution and threat map. So I'm just going to briefly touch on the current distribution map. We're talking about two different maps here. One is the distribution map, and the second map is a threat map, which is a risk map. So the distribution map is based on presence absence data. It is the current distribution of the beetles, specifically Kuroshio and Polyphagous shothole borer. So this doesn't include greater shothole borer currently on the map, but it is based on the survey data and where the beetles have been confirmed and where they have been confirmed to be present and where they've been confirmed to be absent. Okay, so it's based on survey data. So the map can be found here at this website. It's at the UCANR Invasive Shothole Borers website, ISHB FD Distribution, California. And the scroll's like the new way. I'm totally old school. Okay, so here's where the distribution map is. And it's basically, again, it's a present or absent map. So it's showing positive and negative locations represented by different colored squares. And includes beetles identified to the genus or species level. And okay, sorry, got it. So I just wanted to show here, if you go to the website, so there's the two different maps. There's the distribution map here, and then there's the statewide risk map or the threat map. So you can find both different maps here, okay? So here is, if you click here, you can go to the full screen of the map, and this shows the positive and the negative confirmed locations of where the beetle is and is not, okay? So Are there any questions about that? And I can zoom in.
Shannon,
Yes,
those are based on positive identification?
Yes.
Or testing?
positive identification based on trap surveys and positive identification of the fungus. So beetle or fungus, yeah.
Lab testing for the fungus.
No, it can also be based on the beetle identification.
Shannon, can you please repeat the questions coming from the room?
Yes,
There was-
absolutely. The
Thank
question
you.
was the identification based on the fungus, and the answer is it's based on either the beetle or the fungal presence. Any other questions about that? Sweet. Okay, so now I'm going to talk about the threat map, but I want to talk about first how it was made and developed before I talk about how to use it, just so you kind of have like a little bit of a background of the kind of data that went into making it, so that you understand how to use the map itself. Okay, so can everybody see the online the presentation mode? Yeah, okay, great. Okay, so we know that the I'm going to be just talking about Polyphagous and Corrosion Shothole borer now. We know that the beetles have a broad host range that John talked about. And we also know, based on previous work, that the host range, like there is this interesting thing about host ranges where that we understand from an evolutionary perspective, that more closely related species are more likely to share pathogens and pests. And because of that, we can do some really interesting predictive ecology. And so I wanted to use that based on the host range in this system to try and understand if we can use that to predict where the beetle is most likely to go and cause the most damage in this system. And the other thing that we also know is that there's variation in impact within the host range. So in some cases, when the beetle attacks, it only causes branch dieback, but in other cases, it causes actual tree death. So there's a subset of tree species where the trees are only, declining, and in other cases, the trees are actually killed when they're attacked. And John broke this down pretty well, so I'm not going to get into it too much, but there's, a large set of tree species, over 300, where the beetle will make an attempted attack and the fungus cannot colonize on that tree. But then on a subset of those, roughly 100 or so, the fungus can colonize, but the beetle cannot establish a gallery. So the fungus can colonize, but the beetle cannot reproduce. The fungus can't spread from tree to tree, but it can colonize. And then on a much smaller subset of those trees, the beetle can establish a gallery, establish its fungal garden, and then reproduce. And what we call those reproductive hosts or competent hosts.
We once again lost our recording for several slides, so I'll briefly go through what Shannon covered. What we found was that these host types are based on nested types of outcomes and interactions between host plants, the beetles, and the fungal pathogens, where the beetle makes an attempted attack on 324 species, 137 of which only the fungus can colonize, and a subset of 77 species that are competent hosts that support reproduction of fungus and beetles and are important in the spread, including 20 native to California that dominate the landscape, avocado, and anywhere from 25 to 60% of all individuals planted in the urban landscape in Southern California. But a subset of 18 species can be killed when they are attacked. So the lethality of different hosts is important in terms of ecological impact. And what Shannon wanted to know was if these subsets are arbitrary, or if there is predictability in which tree species are attacked and colonized and competent or killed. And because Fusarium dieback hosts are distributed over a complex landscape, where some sites have one species, like an avocado grove, and others have a lot, like in these natural forests, we can use this evolutionary understanding of host relationships to predict in which locations the beetle pathogen is most likely to establish and cause damage in California. To do this, Shannon's team established a large plot network in infested and non-infested locations in peri-urban and wildland forests and avocado groves. To incorporate the whole range of environmental conditions, they collected microclimate data in each and measured the amount of disease, if present, on each tree species. They looked at how vulnerable a site might be to invasion by considering two things together. One, how closely related the plants are at the site are known to host species, and how many of those plants are present. For each plant species in the community, they estimated the chance that it could be a host by measuring how closely it is related evolutionarily to species they already knew are hosts. Then they adjusted the chance depending on how common that plant is in the site. In short, a site is more susceptible if it has lots of plants that are closely related to known hosts. They estimated susceptibility for 170 cities in California, which have complete street inventories in the California Urban Forest Inventory. This represents the largest database of individual urban trees in the United States. Individual tree data of over 5 million trees from the inventory were aggregated to 9,430 1-kilometer squared grids across the extent of California. This is what it looks like when we zoom in. And then the likelihood of FD ISHB establishment was estimated using the model and the total number of trees representing each species for 1 squared kilometer grids. Fortuitously, they had a subset of 832 grids that were not part of model development that they used to test how well the model could predict establishment. And they found that the Philo EPI model predicted observed infestation to grids moderately well.
Okay. So we did have the microclimate data in our plots, and using the microclimate data in our plots, we were also able to estimate the number of generations that each of our monitoring plots could produce within a calendar year. And based off of that, we used DAMA data from online and the parameters from this degree day model, and we estimated the number of beetle generations that could occur within these square kilometer grids across the entire state. So we have this combination of the tree inventory data plus the microclimate data to produce this bioclimatic map across the entire state that can estimate site susceptibility as well. So that is what you're looking at to kind of fill in the grids for the rest of the map. And so this is what the entire risk map looks like when you look at that as a whole. So the sites from yellow to red, those are those clusters there that are based on the street tree inventories. And then you have the sites from yellow to blue that represent the number of beetle generations that could occur within a square kilometer grid. So just by kind of looking at this map, the Central Valley is more blue. So it's basically warmer locations that can produce more beetle generations are going to be more favorable to beetle establishment. Okay, so blue is going to be more favorable. Yellow is going to be less favorable, or light green is going to be less favorable. The interesting thing is that when you look at these in combination, what we found was that the host information is actually more important than microclimate. So it's better when you have those data together to look at. And that's why we really want that host data first. But the climate data is a good first pass to say, okay, you know, the beetle can establish here, but we really want to know what the hosts are in that location as well. So this is just kind of giving some information that when we zoom into some of these sites, this is within the infested range. This is in Ventura, this is Orange County, and this is San Diego County. And this is now infested Santa Clara County right here. Well, actually, this is the Bay Area. Here's Santa Clara County right here. And so what's interesting is it's kind of like in that green area, but you have these areas that are dark, that have like these dark red, which indicate some hot spots for hosts. Does that make sense? But then what's interesting is that this is Sacramento County. It's much darker blue. And then if you can see here, there's a lot of areas that are dark orange to dark red, which indicate hot spots that are highly susceptible that host-wise. And so Sacramento is really an area that we should be monitoring heavily. So that's kind of like how you can use the map to kind of gauge where the areas are going to be more risky than others. So that is essentially how we developed the risk map. What we did was my colleague from CSUMB, John Dedka, turned this into an interactive tool online, and that is the threat map. So the risk map is online. It explains everything that I said, but much more concisely. Okay.
The easy URL to go to though is ishb.org.
Go to ishb.org and you can find the map here. So there's an explanation of that. There's also a video tutorial that explains how to use the interactive map as well. So you can click on that and watch a 6-minute video tutorial that explains how to use the map. So again, it's based on tree composition and climate conditions. The map is here, but I'm going to click here to view it in full screen now.
Shannon, there's one online question that's 2 parts about this section before you transition to the next topic.
About this section, okay?
Right. Do you want it now, or you want to talk about that, the risk map?
I can, sure, go ahead.
Okay, I'm going to give you both parts simultaneously. First part is, what is the equation for calculating the number of generations, and is it only temperature dependent?
Yes, okay. The equation is, yeah, what is the equation for calculating? I feel like I'm on my like PhD defense or something. What's the equation for calculating the degree days? It's a complicated equation that's based on that's based on like the sign of the minimum and maximum temperature of the growth parameters of the beetles the growth constraints of the beetles. So it's based on temperature only.
Shannon, so can you use the IPM degree model online?
This one is better.
I think that's-
Can you,
Wait.
the question was, can you use the IPM degree day model online? I haven't, I can try and see how it compares. But this one, you know, was run through several lines of code, and it was good.
Can you put something like that online? Like on the IPM website or ISHP website?
We can. I'll go through that actually. After I go through this map, I'll go through how we want to improve this map and things like that. Great question. Is there another, so the second part to that question? Julie?
They were given simultaneous. You're done with that question.
Okay, I'm done with that question. Okay.
Yeah.
All right. Okay, so here is the map. And basically what it's showing right now is the distribution of risk based on the host tree composition. This is showing here right now in full view a panel, okay? And on the panel, I wonder if I can, no, it won't show. Okay. The panel is not, I don't know how to unzoom this and make it smaller. Maybe if I do this. Oh, is it command, not control? I tried control negative, but it's not moving. Oh, look at that simple zoom out. No, that's not what I failed my defense. No, that's not what I was after. Actually, I want to make this whole thing smaller. But anyway, it does have a thing for the degree days as well on here. But basically, this panel is showing you can search by county, so you can actually type in the search, or there's a drop-down menu where you can just like click on the county that you want to zoom into. And zoom into that county. You can also use this on your smart device, so on your iPad or your iPhone, and you can click on the location that you are at currently and zoom into your current location. Let's just do this time. And so, look, here we are. So that's kind of a nifty thing there. Okay. Now I'm going to, this can be bulky and it can block like your entire field of view. So I'm gonna hide the panel. And so now I can see a larger portion of the map as well. Yeah. Up here are the layers. So I'm going to turn on the ISHB generations, and then it's going to turn on that generation layer and fill in the rest of the map. So now we've got the host and the generation layer. Now, what's nice is behind this is that satellite map. You can also change it to terrain, but I like the satellite personally. So we want to be able to see where we are, like if we're on our smart device and stuff and we're doing monitoring efforts. So here is this nifty little toggle. So on host related risk, what I'm going to do is I'm going to turn this down, and I'm going to turn off host generations for now. So you can make this more transparent, but still see the amount of risk that's within each block and see where you are in space as well. So that helps to kind of, when you're on the ground and you're putting in your traps, or you want to do any kind of management efforts or whatever you're planning, you can see the actual location in space and see what the risk is in that space as well.
Does it work on Android?
Yeah, it works on any smart device.
But Android is a device for smart people.
Yes. So that's essentially how you use the map. So it's useful to be able to have that transparency, to be able to see your location and also see what the risk is in that location as well. And that's essentially it. We, one of the things that, like what John was alluding to, what we would like to do to improve upon this map. So you saw what goes into this map. One of the things that we're working on is creating a couple of things. We're going to be doing some modeling, additional modeling to incorporate how the landscape factors, landscape characteristics factor into the risk at those particular sites, because that needs to get factored into the model. We also want to incorporate more inventories into this map. And so one of the things that we would like to develop in collaboration with stakeholders is this ability for stakeholders to input their data into the map, and so that it is basically running the model in the background and then continually updating the map in real time. So that is one of the things that we would like to develop and work on. And so it's incorporating new data constantly, including climate data that we can import from the from updated weather stations and things like that, to continuously like update the degree day information as well. So tree inventory data continuously updated and climate data continuously updated. So those are some of the things that we would like to work on with stakeholders. So this map is not perfect. It's a tool, but it is an excellent first pass to help you gauge. Don't use it as like a hard, you know, fast rule, like, you know, this is it. This is like, this is gonna tell me exactly, you know, where things are and what's going on, but it's gonna give you a good measure of where risk is and where the risk is less. Any questions?
I have a question.
Yes.
So earlier you mentioned the monitoring, that perhaps the greater shothole borer
Yes.
might need greater concentration. Was it a hormonal lure, or is it something else?
We believe it's based on, just because it's functionally similar, behaves functionally the same as Borers-Fusarium and Prolificus shothole borer. We think it's going to be something like a Corsiverol based lure, because it uses the fungi that are in the same group as the fungi that are, you know, the invasive shothole borer species use. Corsiverol is based on the cues that are used by the fungus.
So is that substance, is it terpene related, or is it hormonal?
No, it's based on it's based on the fungus. Yeah.
And then you also mentioned Was it no choice testing?
Yes.
No choice testing.
Okay, yeah, good question. Thank you. So it's basically
Excuse me,
where you-
Shannon, Shannon,
Yeah,
can you
sorry,
please
sorry, sorry,
repeat?
sorry. The question
That's
was,
okay.
the first question was about, what kind of chemical was the lure based on that we were gonna be doing the optimizing on for the traps for the greater shothole borer? And the second question was, what are host choice testing? What's host choice testing? No choice testing. What is no choice testing? So no choice testing is basically where you take the beetle and you basically give it, you introduce it to the tree and you don't give it an option to try to invade like another tree species. So basically we take we put it into like a trap and we put that trap onto a host, like a maple, and it can't escape that trap. So it just like hangs out. So it either is going to die in that trap or it's going to try and produce a gallery into the maple. If it produces a gallery into the maple, then it's like, okay, it can establish a gallery in maple. It had no choice but to establish a gallery. If it doesn't establish a gallery and it just dies in the trap, then that means that it is not a host.
What are the temperature constraints? We expected to see them really north, or freezing.
Yeah, the question is, what are the temperature constraints of the beetle? So the beetle cannot handle freezing temperatures. The constraint, I can't remember, do you remember, Paul, the minimum and maximum temperatures? I don't remember off the top of my head. Between what?
70 and 90.
Between 70 and 90 degrees, yeah. So that's why on this map,
But actually, we will survive those 70 degrees.
the Sierra Nevada is basically not a very susceptible location. That's why it's yellow, because you get snow. What's that? Yes, I'm done, unless there's any more questions. I'll get this.
So our next presenter is Bea. We don't know her. She's University of California Cooperative Extension Advisor at Oak Knoll Village counties, specializing in urban forestry. Research and extension program is focused on finding management strategies for urban forest pests, such as invasive shothole borers and multiplier borer.
She's also on the SciSac committee.
Yes, and
That
she
is
is
correct.
the co-chair for the invasive shothole borer subcommittee of SciSac.
Yes. Thank you. And the other co-chair is Shannon. Okay, hopefully this is working. Hi. So the part that I want to cover is management, and so let's get to it. No, this is not working, so bloom or something. Come on. Okay, so I'm gonna ask you guys, what are the main components of a management plan?
Monitoring.
Monitoring, great. What else? There's coffee outside? What? You have to have the pest, yes, so you have to know that you have the right pest. Perfect. What else? Treatments, yes, treatments. Inventory, yay. Okay, so as Kelly will say, you need an accurate identification. We want regular monitoring and assessment. We need clear thresholds for action also, right? So we're gonna do treatments when, why, and of course we need to have a good array of management tactics that we can use. Who remembers what are the IPM different tactics, the three groups? Hey, nice. Who remembers the 3 IPM kinds of tactics? I'm gonna put you in a test. You put Shannon, you put my girl into a test. I'm gonna put you in a test. Who remembers? Biological control, cultural, chemical, yay. All right. And of course, we always need ongoing evaluation. We cannot just do something and then move on. We need to figure out if it worked, what do we need to tweak to get this going, right? But something that I find is that monitoring is actually the backbone. You cannot do an effective management if you don't know what needs to be treated, what doesn't need to be treated, and when. So all the programs I've seen that are actually working and are actually reducing the number of beetles in their locations have a big investment in monitoring. I can say 2 out of the top of my head. Disneyland, right? Disneyland is, yes, Disneyland has a lot of funds to invest in getting Keith and Shannon and John to go and check on the trees. But the main thing that they do is that they survey their trees very often, and they just spend a lot of effort in the monitoring part. And so then they can make good decisions about, okay, are we treating these trees or not? Is this tree passing the threshold or not? Does this tree recover? Or like, is it improving? Or do we need to do something else? Another example is Orange County Parks. Orange County Parks, and that's, you know, they manage more than 50,000 trees, right? Yet at least once a year, every tree in those turf parks get, you know, assessed by those guys over there, Jan and Chris, and a few years ago, they or other Chris, right? So, and that makes a big difference again, because we make the right choice, we save money in the end by looking at, you know, making the decisions, the right decisions for the right tree. So I cannot emphasize this enough. Monitoring should be the first thing that should jump to your idea of managing invasive shothole borers. You will save, in the end, a lot of money if you put first a monitoring plan going on. All right, so how do we monitor? One option is trapping. We know trapping is not good for control, but it's good for knowing what's where. We have these 2 kinds of traps. We have the lingering funnel traps or the sticky funnel trap. We've been switched to using this ones, but they're simpler to use. As Shannon was mentioning, there's Jen has a sticky funnel trap on her hand, and later on we can pass it on. It has beetles in it. So we use Fusarium as a lure. The lure is very mild, attractive in a way. So if we put a trap with a lure, there's no risk that we're going to be attracting beetles from all the areas around, like far away from that place. We're not attracting beetles to our property or the property we're managing. We're only going to be attracting the beetles that are already in the vicinity of the trap. So there's no risk of bringing beetles into your location. If you put a trap, you're actually going to catch what's there, but also you're only going to catch what's, you know, within maybe 50, 100 feet from that trap. So it gives you a very pinpoint picture of what's going on there. We like to use them a lot in combination with visual surveys, and we use them to check where do we need to look more closely. And what happens a lot of the times, and I'm gonna give you an example in Orange County Parks and Irvine Regional Park. We keep seeing around the zoo, the sycamores kept getting infested. We were treating and removing and doing all the things that I'm gonna talk to you about, and yet the sycamores kept getting reinfested. And we couldn't figure out where it was coming from. And we started putting traps, and we figured out it was those huge coast-like oaks by the zoo, which are also another hard thing to look at because a lot of them are in or next to enclosures. But the beetles were having colonies in the top branches of those oaks. And we wouldn't know that if we didn't have the traps, because the traps were saying, no, no, something is going on in here, go look closely, right? And especially in situations where you can't have a lifter to go check on all the high branches, it's good to have this thing where it's like, okay, well, I can have a lifter for a couple of hours maybe. Where? Okay, there, in that location. And then we go and check on those branches, and then we make decisions based on that. So it's really useful to do both. Trapping and also visual monitoring. In trapping also, it's useful to look at the yearly patterns of flight for these beetles. So in general, we have two sort of peaks in the timing that these beetles fly. One tends to be in spring, and the other one tends to be in the fall. And it kind of varies depending on the year and depending on the location. Here you have two Orange County parks, Ladera Ranch and Irvine. And there, this is a combination of many years. But as you can see, in Ladera Ranch, the spring peak ends up being like May, versus in Irvine, it tends to be like in February. And the fall peak kind of coincide in both of them. But another interesting thing is that in Irvine, it seems like the fall peak is a lot stronger than the spring peak. And so knowing these things for your specific location would really help you time the treatments, if you decide to do so, at the best moment for your location, right? If you're going to be doing any kind of contact insecticide sprays in Irvine, I would recommend to do it in the fall because that's when they are flying the most, right? And if you're gonna do it in Ladera Ranch, you could do it in the fall, or you can do it maybe like in April, May when you have a peak. But if you say, oh, I'm gonna do it the same like in February, March when there's a peak in Irvine, you're kind of applying the treatment before you actually have a lot before you have the peak, and by the time you have the peak, maybe at least half of a lot of that product degraded already. So really interesting and really important to use this as part of your management. Another point towards monitoring. It always pair that with visual inspections. That is the way you're actually going to collect data that's going to allow you to make management decisions at the individual tree level. So what we do is we observe the whole tree from all sides, and we assign one of these levels of infestation: low, moderate, heavy, and severe, based on the number of active nitrogen holes. Less than 50 is low, between 50 and 150 is moderate, more than 150 is heavily infested, and more than 150 in Dieback, that's a severe infested tree. And one thing that you might notice is the number of active entry exit holes. And that's because as infestation progresses, you have two things. One is old holes from old galleries, old infestations that are, those beetles already did their thing and left. But also, as you manage and as you apply some treatments, you're allowing for the tree to respond. It might be compartmentalizing around that gallery and might not be allowing that fungus to proliferate. And so those galleries are no longer have beetles just because the beetles died. And so they are recovered trees. So we have been looking at a lot of trees, and based on a lot of observations, this is kind of the main things that we can do to distinguish active holes versus inactive holes. So holes, active holes, where you have a beetle that's alive in there and it's making more beetles, and inactive holes are holes from an old gallery that no longer has a beetle in it. And the main differences is active holes are oozing, they're wet, you can see grass, sometimes you can even see the abdomen of the beetle sticking out. So all of that is a good indication. It's a good indication that this is an active hole, right? The inactive holes, they kind of look like they're dry. There's no oozing from it. There's no frass coming out. A lot of the times, depending on the species, for example, for London flames, they start bulging like this. Our cat. Took him with that game. It starts bulging like this, and so the entry hole kind of doesn't look round anymore, it looks deformed. And in sycamores, you still see the entry exit hole, but it's dry, it has nothing around it, and it's kind of grayish, this grayish tissue around it. It's a kind of dead tissue. That is the tissue that has been killed by the fungus, but at this point it's compartmentalized and it's just sitting there. So, and we know this because we looked at a lot of these and we tested them. So how do we test if they're active or not? You cover it with paint and you wait maybe a few hours, maybe you come back the next day and you see if it's reopened. The beetle isn't there and she loves her. She will reopen if she's in there. And if it's still closed, that hole is inactive. And we did that with hundreds of holes, and all the holes that looked this didn't reopen. And let's see if this works. Oops, it was working. And this is what it looks like when she starts pushing the paint out to reopen that hole. So if you're lucky, you can see that happening live. So what happens with those trees that only have this inactive holes? We've seen that, especially here in Orange County, where the beetles have been here for a while. These are what we call recovering trees. Yes, there are, because they look pretty good. Their canopy is fine, and they have all these holes, and we painted all of these holes, and none of them reopened. So we know that there's no beetles in there. And so what we notice is that the tree can go from light infested and start progressing to moderate severely infested and eventually be removed, or sometimes it goes this way and recovers. So what I did is started to look, okay, how does that look over time? So I grabbed the inventory that we had from OC parks. We had 10 parks. Only the platinum species of sycamores and London planes, basically, we had more than 5,000 trees. And we looked at what happened through 7 different years of monitoring and inventorying those trees. We learned a lot. So starting here, this is the first, in 2018, the first year that we're looking at this inventory. We see about half of the trees are infested and half of the trees are non-infested. This green is uninfested, never been touched by shothole. In between the infested, most of them are lightly infested, right? And as we progress, we see several trends. One is that because these trees are somewhat managed, the number of infested trees starts shrinking. Of course, a very awful thing that happens is the number of removed trees keep growing, because a lot of those trees did go into the pathway of getting severely infested and had to be removed. Yet, we find this blue category that keeps growing. And these are the trees that actually recovered. It means the trees that at some point were qualified as infested on any category, and later on, they had only inactive holes. So this is showing us that there is a path in which you can have a lot of the trees that are recovered. So they're now considered uninfested. And yes, you're gonna lose a lot of trees, right? But there is a path towards something that now looks like of the remaining trees, most of them are uninfested, either recovered or have never been infested. And those, to me, are good news, that with good management, we can get something like that. Now, I looked at this and put it into this amazing matrix, and I'm gonna explain it a little bit. So this is a way of looking, based on the current state of the tree, this y-axis, what's the probability that the next year you'll come and we'll find it in the next state, which is the x-axis, right? So, since it's uninfested this year, what is the probability that the next year will be uninfested? And the probability is 81.7%. Now, what is the probability for an uninfested tree to become heavily infested the next year? 0.2%, and so on. So, this diagonal is showing the probability of staying within the same pattern, right? Some things are obvious. If a tree has been removed, the next year it doesn't come back. It's being removed. So that makes sense. 100% here makes sense. And so what is showing us in here is, for example, for lightly infested trees, the chance of remaining lightly infested is pretty high. It's like 57%. And it also has a pretty high chance of recovering, like 37%. That is really good. And moderately infested trees also have a pretty high chance of getting of being lightly infested next time or recover next time. So they might stay in the same category with 3% chance, or they might drop to low, that's another 33%, or might drop to uninfested, that's 32%. That is really good. So that's a good news for us thinking about tree recovery. Now, once we start to the heavily infested tree, the situation looks a little bit different. So heavily infested tree, the highest possibility, the highest probability for the next state is meaning it just got too bad, and by the next year it had to be removed. And same thing for severe, right? Yet there's still a non-equal to 0 plus chance that a heavily infested tree might recover, or even a severely infested tree. So for those very high value trees that you want to invest a lot into maybe treating them, there is a chance, about 10%, a little bit less than 10%, that that tree might recover. So this shows us that uninfested is very stable. Like most uninfested trees stay uninfested. That's really good. Low and moderate is kind of categories that will show high probability of improvement, right? Just moving towards uninfested. Heavy is a tipping point. Heavily infested trees tend to progress most towards being severely infested trees and eventually remove. And severe, you have very low chance of recovery. So, with this, we learned that there is a chance for recovery, and we have to, and we can make decisions based on that, right? And we can just maybe put our efforts into this moderately infested trees if we wanted to kind of treat this.
These are treated trees.
This is a mixture. Okay. This is more than 5,000 trees in 10 parks. Some of them were treated, some of them were not. But in all of those parks, the amplifier trees were removed quite soon. I still have to dig into this data a little bit more to see if I can extract this treated versus untreated. So, what are our options now if we want to do something with these trees, right? So, as we were talking about, our options are the cultural, physical, mechanical, chemical, and biological control. So let's go to cultural. We can prune the infested branches. Every time the infestation is located mostly in a branch, we can prune that branch. We're just removing those beetles out of the equation. We can remove amplifier trees, which we know is a very good way of removing a lot of trees, a lot of beetles out of that. Also removing a lot of trees, but I meant a lot of beetles out of that area. We don't want to remove a lot of trees. And then we need to think about what do we do with that infested material. So the severely infested trees, they are not only have low probability of recovering, but they're also hazard trees. The branches that are heavily infested have all these galleries in them. There's a lot of structural compromise of the tissue, and so they tend to get brittle and break. So you don't want that falling on someone's head or someone's car. And so these trees, the heavily infested trees, we want to remove them quickly. And also we want to make sure that the stump gets removed, especially if we're talking about turf area where you can actually bring a stump grinder or something like that. And the reason why is because we still have holes in those stumps, and those stumps can still pump out beetles. Now, if you are managing an area that is riparian area or wilderness area, you might want to keep the stumps, maybe just cut it as flush to the ground as possible, because you want that resprouting to happen, especially if you're managing willows or sycamores, they can resprout pretty nicely. And then for when you have infestations in the branches, you can just remove those branches, and then you don't have to go at that tree with everything you have. So in general, avocados, all we do is remove branches. Oaks, especially coast live oak, is not a very preferred host, but it's a very common host that gets everywhere. And so they end up in those high branches. And so we can remove those high branches and get rid of a lot of the beetles without having to do a lot of treatments. But now what happens with, these branches have a lot, each of these savings is a beetle, the entrance of a beetle gallery. So we know that these beetles can live in downwood for a long, long time. So we want to prevent the spread. So what we want to do is manage this infested plant material so we are not spreading the pest somewhere else. Our options is to chip it. Chip it as small as possible. We always say chip to less than 1 inch. I know that is very, very hard to attain, but as small as possible is chipping is better than not chipping for sure. Even if the chips you can get are like 2 inches big or 5 inches big, chipping is always better. But those chips, especially if they're not 1 inch or smaller, can still harbor some beetles. So we could use them in the same property as mulch, but definitely don't move them to another property because you could be moving beetles with those chips. Ideally, you just take those chips to a composting facility, and once the composting happens as it should be pest-free after that process. And please don't move firewood. No matter how tempted it is to put a bunch of firewood in your convertible, just don't do it. Because that way you're moving not just shothole, but any other, pest that move in firewood. All right, so now to our chemical options. We have trunk sprays. The ones that we know that have been tested and have some effect on the beetle population are bifenthrin, that's an contact insecticide, and we usually mix that with Bacillus subtilis, which is a biological fungicide. You can mix both of them together and spray them, that is okay, I asked. And then what you want to do is apply this right before they start dispersing. Again, that's why a trapping program is really good, because then you will know in your location when the peak happens, and you can apply this right before that. You will spray this just in the trunks and the branches, no spraying in the foliage is needed. And then, of course, because the kind of insecticide that is, you should avoid doing this close to any flowering plants or bodies of water. Other options are systemic pesticides. So in this case, we depend on the tree to actually uptake that pesticide and bring it up. And so ideally you would irrigate that tree before the treatment, and you could do a soil injection or soil drench with imidacloprid. That works pretty good, but if the soil is impacted or you don't have a good irrigation, that's not gonna work that well. And also because it's a soil injection, it could leach down the soil and into bodies of water. So you don't want to do soil injection close to bodies of water either. And that leaves you with the last option, which is the trunk injection. In this case, the pesticides that we have is imamectin benzoate, which is an insecticide, and propiconazole, which is a fungicide. We're trying to kick them from both sides, right? The fungi and the beetle. You can also inject both of them together, but when you do a trunk injection, you're drilling into the trunk. And unfortunately, especially here in Southern California, there's a lot of bud canker spores. We keep seeing this injection sites getting infested with other pathogens, and then we end up having a worse problem. So for trunk injection, sometimes that's all you can do, and that's fine. Just think about the pros and cons, and if you're talking to a client, you need to, share that with the client. This is the good things about trunk injection is that you're just keeping everything inside the trunk, but there's a chance that they could get infested, and that makes a worse problem. One thing we do for this is we use that same Bacillus subtilis biological fungicide, and what we do is we mix that and we have it in a spray bottle. And then after we put our teeth and we do the injection, we finish the injection, we take all the equipment out, and in each of those holes, we spray with the Bacillus subtilis solution into the hole and around the hole. And we do that in all the injection holes. And that tends to help prevent a little bit that possibility of fungal infection after. Yes?
Go up to a canker.
Yes, so John, I'm just gonna repeat for online, what John was referring to is when you're doing the drills, let's say you're drilling in your spot and turns out that that spot had a canker underneath, and you'll notice that because the frass that, the drill sawdust that comes out of the drill, instead of being light color, it's dark in color. So you drill into an area that is already infested with some kind of canker. One, you cannot inject there because that tissue is not going to be taking up anything because it's dead or dying. And two, you should not use that drill to keep making drills around in that tree or in other trees. That drill bit is infested. So you should switch the drill bit or disinfect it before you continue working. We talked about disinfection. You can use, I like to use ethanol, like alcohol, at 70% because it runs nicely and it doesn't rust my blades. But you can also use bleach at 5%, though it will rust anything that is metal. Or you can use Lysol to spray it. But ideally, you will have several drill bits, and whenever you end up in a canker situation, you change the drill bit and you can deal with disinfecting later. Thank you, John. All right, and now last option, and this is fairly new, is spot injection. So this is basically grabbing a syringe and injecting literally a droplet of pesticide directly into the entry hole of the beetle, and it feels great. But it takes a lot of work. So this is something that we know it works. We tried it with acephate, imamectin benzoate, and imidacloprid, and both imamectin benzoate and imidacloprid showed really good control compared with the control. but it's very labor intensive. So it's something to consider for special situations, maybe high value trees. High value tree, again, what is a high value tree? It just depends. If it's my backyard and I have 2 trees, both of them are high value trees, and they have 5 exit holes, and I want to make sure they die, we can do this. It's just something else to have in your toolbox depending on the situation, but it is pretty labor intensive. The good thing is that it uses 0.1 or to 0.2 milliliters. This is literally 1 droplet. And John, yes.
You're lucky you can impale a beetle.
So bottom line, if you're doing treatments, what are you going to do as a treatment depends on the situation. Like in this case, you're next to a body of water, your only option is to do a trunk injection. And in this case, if you want to do a soil injection, you probably are out of luck because there's almost no soil in there, but it's a parking lot, so it's pretty good to do a trunk spray. So these decisions have to be done in a tree by tree basis. So considerations, there's no preventative treatment, so only consider treating trees that are infested. If the tree is not infested, just leave it alone, basically. We haven't had any evidence that preventative treatments really work, and you could be doing more damage than good. Generally, we reserve this for moderately or heavily infested trees. I am trying to get funding to study what happens if we only treat these trees and we leave the lightly infested trees alone, and if those lightly infested trees, when you have everything else managed, might actually recover on their own. This is something that we've been seeing, but yeah, send money right away. You must be a licensed applicant, pesticide applicator to use this treatments, and remember that no treatment works 100%, so you actually have to go back and check what happened, and you might have to change your strategy depending on what you see next year. For biological control, there's current research going on, and I'm not going to dive into that because we have talks later on today that are going to tackle that. And so this is when we talked about making a decision. This is something that will help you, but still remember it's a tree by tree basis. But this kind of matrix is showing based on the type of host, if it's reproductive or not, the hazard level, and the level of infestation, what will your action or recommended action be? And this is for low value trees or high value trees. And it's basically sort of condensating all the information that I just showed you. So I'm not going to dive into this too much. Just know that you can find this at isnt.org. In this this publication has that matrix in there. If you wanna, you can take a picture with your phone and have it right now if you want. Or you go browse our website where you can find the map that Shannon showed. So you should not forget isnt.org. All right, so last thing. Once we're removing so many trees, we need to think about replanting. And so what things are we gonna be considering when we think about what are we gonna be replanting? So the first one is the type of habitat. Is it urban or is it a natural forest, a riparian forest? When we're replanting in a natural habitat, we are very limited on what we can replant. And unfortunately, most of the hosts, or almost all of the hosts, all of the trees that naturally occur here, at least in Southern California, in riparian habitats, are hosts for shothole. So your palette is getting a lot restricted. There's some light at the end of the tunnel that Shannon and Akif are gonna tell us about, so stay tuned. But if you have an urban area, you have a lot more options. So you need to think about also what the infestation level is in the area. Are you gonna replace that sycamore with another sycamore? Well, have you treated or managed the other sycamores? Is the area heavily infested? Then please don't plant another host because it's gonna get infested. You still have some grace period because these beetles kind of like trees that are bigger, like bigger to the age. So you have some time to manage your current infestation, but think about that when you're making those decisions. You know, you don't have to say, oh, I'm not planting hosts, because if your area is well managed, you can replant with hosts. But then think about what's already there, right? We all learned from the Santa Moore's rule, the 10, 20, 30, and that's that nice guidance, right? You have to think about, if you're gonna think about what to put in an area, you don't want any more than 10% of the trees to be the same species, or 20% of them to be the same genus, or 30% of them to be the same family. And as Shannon was showing, the hosts that showers like tend to be closely related. So you have to have your inventories in place to know what you have, and then you can decide what to plant, aiming for, you know, if you have a street or a park that's 25% sycamores, don't plant another sycamore, right? Aim for diversity. It has been, in, you know, many research has been shown that when you have more diversity, the occurrence of pests tend to drop. Like, diverse systems are more resilient, and that works with total species diversity, or the evolutionary distance from, you know, of all the host species, which is kind of what Shannon was looking at with all the hosts together. So you want to have an urban forest that is as diverse as possible, so it's as resilient as possible. Don't, you know, another thing is like, oh, I'm just gonna plant for another host, so all of these are gonna be pines. And then you have the pine ghost canker killing all your pines, right? So always aim for diversity. And lastly, remember, and I mean, most of you are arborists, but right tree for the right place, right? Think you have now an option that maybe a manager before you made a really bad decision, planted a tree that grew too high and there's a power line or whatever in there. Well, now you have an option. Now you can decide what kind of tree goes there, how high that tree is gonna grow, how the root system of that tree is, what are the needs on the soil and irrigation of that tree. You can make those right decisions right now. And you can use the select tree tool made by Cal Poly to give you an idea of what options you have. And with that, conclusions. Keep calm on follow IPM. Shothole borer is here to stay, but no, we don't need to panic. We can manage it. We just need to educate ourselves about shothole borer and other pests that might be affecting the tree. Make sure we know how to identify it. We can make take preventative actions so we don't spread the pests. We look for science-based management options like the ones I show you, and then just look at all the tools you have in your toolbox before you decide to use pesticides to manage those trees. And that's it. Thank you.
Hi, Bea, we have one online question.
We do have one question online.
Okay.
Somebody's asking if there's a list of pesticide applicators who are knowledgeable about ISHB treatment.
So there's someone asking if there's a list of applicators that are knowledgeable about shothole borers and shothole borer treatments. We don't keep tabs on the people. I mean, we keep a list of who comes, but we don't make that to our trainings. We don't make that available. So what you can do is make sure that your arborist is ISA certified, and then you can ask those questions. Have you been in a training for shothole borer? You know what shothole borer is? Can you show me which training did you go? And you know, ask questions and see if the professional you're considering actually has some training, but we don't have any specific list. Here in the audience, we have one, Tim.
So have you seen any, or has anybody done research about when the larvae mature into adults, and they're ready to go up into the tree elsewhere and reproduce. Are they coming out of the same hole that was, like you said, painted, right? Are they coming out of that same hole, or are they boring out of the tree and then boring back in? Or is it like a mix? Like, how does that actually happen?
All right, good question. I'm gonna repeat it for the online people. So when a larvae develops into a new adult and is ready to go and disperse, that's it, go out of the gallery on the same entry hole where the mother basically made that gallery, or do they bore another exit hole? And as long as we know, as far as we know, it's probably the same. Sometimes the same hole, so that's why we call it entry-exit hole. In some other pests, you hear, oh, exit hole. And some pests have the entry hole and the exit hole look different. But in this case, in most cases, it's the same one. Sometimes the galleries get a little bit out of control and they start just like merging with each other. So maybe it came in on one and ends up living on auntie beetle a little bit far away, but it generally is the same. Yes. Yeah.
I have kind of a related question to that. Has anyone tried like plugging holes? Like might be like a low tech, not chemical way to kind of trap them inside.
Just with a few things, and in general, they're pretty good at making that hole open again. I mean, they can chew through wood, so there's very little things that they can chew through plastic very easily. So far we haven't found anything that traps them there. All right. Oh, Rachel.
I just have a question on the recovering trees. So the photo that you showed, the recovering trees had all the holes been active. It would have been, at least heavily infested to amplify the tree.
Yeah.
What happens if you were to cut the recovering tree? Are all the galleries inside also recovering and sealing up, or is there any kind of leftover weakness in part of the technique after it recovers?
Yeah, that's a great question. So Rachel was asking, in those recovered trees, what happens inside? Does it look recovered inside? Are those galleries don't harbor any fungus anymore, or is there a weakness to that tree? We don't have much data on recovered trees that have been cut because we just keep them for the most part. But what I can tell you is there might be a little bit of a structural weakness, but it doesn't show much. And also the canopy looks pretty good. The other thing that we did is we took samples of like core samples of those trees right where the holes of the galleries were, just a little bit higher than that. So just to reach the cambium and basically see if there was fungus active in there. And we didn't recover any Fusarium from that. We did recover a little bit of Fusarium from the gallery entrance. So there might be some in there sort of holding up, but it doesn't seem to be affecting the tree much. And the other thing that we checked is if those trees were maybe more attractive to beetles, and we didn't find any evidence that they were. We placed traps stapled to those trees and to healthy trees, and we didn't find any difference. So it seems like they're fine. Yes. I think.
So our next speaker is Kelly Parkin. She is the plant health care coordinator for West Coast Arborists. And she joined the company in 2021. Career endeavors over the last 42 years include work in horticulture, golf, and sports turf, and park maintenance in the municipal sector. She's a licensed pest control advisor, a certified arborist, and a certified crop advisor. She has a bachelor's degree in biology from the University of Delaware and a master's degree in plant science from Cal Poly Pomona. Her position as PHC coordinator includes program development, site-specific tree monitoring, pesticide treatment recommendations, and client consultation. Take it away, Kelly.
Good morning. So Bear pretty much gave my whole presentation. So She did a much better job. I've been asked to look at some best management practices. I have a little bit of redundancy on mine, so I'll go quickly through those and then look at some of the harder stuff. So I am going to give a perspective on the challenge of ISHB. Our tools and our partnerships are pretty much our inventory system and our inventory specialists. We do quite a bit of monitoring. Our monitoring is not trapping per se. We really use cooperative extension information on that. Ours is more survey out in the field with our inventory specialists and our applicators and PCA, who's me. We use the ECC guidelines for monitoring and treatment and for the employment and treatment protocols. So I'm going to really spend a lot of time here with the obstacles and workarounds. It always sounds great sitting in these seminars going, yeah, you know, that should work out great. You know, you go out into the field and it's like, why is that dead? You know, I thought that program was going to work. So there's a lot of things that go into getting these treatments to work. Most of it, from our perspective, is good follow-up. If we can get out there in a quick turnaround and kind of see what's going on out there, then we have a pretty good chance of continuing to treat healthy trees. So again, effectiveness of the treatment, and then the pesticide regulation, public perception. I'm going to go through a little bit of that at the end if I have some time. So my job is a coordinator position for West Coast Arborists. I'm the PCA essentially for public agencies. That's primarily our client. We've been treating and evaluating thousands of trees since 2017. So we have a pretty good idea on some of the programs that we've implemented and the results that we've got. We've been in Southern California for those 8 years. The Northern California pest was identified last year. We're part of the working group, but we've not done any of the programs I'm going to describe to you today up in Northern California. We feel like we will, but we have not done that yet. And then I kind of look at how many trees became an amplifier that were on our program. Maybe that's not scientific or that's not the right way to look at it, but we've lost very few trees. I think that's the takeaway message, is that we've lost very few trees that have been on a monitoring, treatment, and then evaluation program after that. 2024, we lost trees that have been on the program, and I still don't have a really good idea. I'm very curious about the effect of generational rains for 2 years on the health of the insect and the ability to grow the Fusarium disease. So we had more struggles in 2024. This seemed much better in 2025 on the same program. So still trying to figure out what's going on there. So this gives an idea about just where we're working, primarily Los Angeles and Orange counties. We do have some agencies in San Bernardino, San Diego, Riverside, and Ventura. But most of our work is Los Angeles and Orange County. And then it's kind of same thing there was going through is the IPM management tools that have been developed work very well. We employ them. We educate our clients on how we use them and why we use them. The education part of this is a big deal. I love the maps and everything that Shannon's showing about. I can really see sending our clients to those maps so they can see that they're in an area that is a prime area for this pest. So we're out there evaluating during these monitoring programs, and we're looking for dry hits. Dry hits means the tree is really managing that pest. And we've gone back and looked at some of the trees that we've treated, and there are no live hits, they're all dry hits. We would not take that tree and treat it again if that's all we're finding. Wet hits are a great indicator of an active infestation. Fusarium disease impact on the canopy is primarily what I use to gauge the health of the tree. Is this a tree we're going to treat or not? And then trees with moderate levels, even high levels with full canopies are good candidates. So, you know, the thing we have to remember is the insect is in there, and we have to work with the insect. We're really trying to control that disease and the impact of the disease on the canopy. This matrix is pretty well gone over. I'm going to skip through that. There are management guidelines that Bea went over, and all of these are important, but we have to remember that we don't own these trees. We're a contractor for these public agency clients. So we primarily, when we're monitoring, are looking at this. There are value assessments and hazard levels, but those are risk assessments, right? The value assessment is given by the owner of the tree. We may go out there and tell you got 100 trees on a property that are moderately infested and need to be treated. They're like, we don't care about those trees. Oh, that's pretty tough, because if they don't, if that's a property, like say it's a wildlands area that borders a park and they're more concerned about the park, having the infestation level of the host is one criteria, but they're looking at the value of the trees against mostly budgets. So there is that, there's always that pull when you're looking at these programs. And again, these are not risk assessments. Some of these trees are maybe higher risk level because they are infested, but this would be a different type of evaluation we would do. We would not do this at the same time as we would assess the insect level. So again, we have the inventory system. I'm not going to go over this. Kind of everyone knows what their inventory system is. But the treatments Bea went through are pretty much how we do this. Most of our trees are in parks or street trees. They're right-of-way trees. And we try to monitor right-of-way trees if we have the opportunity. They're in our inventories, because a lot of times those have infestations that continue to spread if we're only doing the park trees. So no infestation, no treatment per the 2023 guidelines. That is not a hard and fast rule. We have treated no infestation trees. We have a park that has 100 trees that are infested, somewhere between, moderately infested or even high infested. We have a handful of trees in that park that are not infested. At times we will elect or recommend to our client to treat the non-infested trees, particularly if we won't be back to see those trees for a long period of time. It's a one-off. I don't know what's wrong with my trees. I want you to come out and look at them. We go out and look at them. You've got ISHB, you've got it at a fairly high level. Well, I got this money this year and that's all I have. Okay, well, we might recommend to them, depending on the level of infestation, that you have low or no infestation trees to treat. These are generally trunk sprays with a bifenthrin, a penetrant of some kind, and then we use an extender with our treatments. We find that, especially in park situations, you have irrigation heads hitting these trunks. We're trying to extend the interval that it's active against the pest. So we generally will include that in, and it's very small cost. I forget what it is, but it's like $4 per 100 gallons, so it's pretty cheap. No infestation. We don't include anything to address the Fusarium disease. Low infestation, again, Bea went through this pretty well, so I'm not going to spend a lot of time, but we almost never use dinotefuran. It's expensive. We use a bifenthrin, a bacillus product like cease, the pentrabark, and the new film. And then we use systemic soil injections with imidacloprid only when the area around the trees appear to be in good condition. So if these trees have very, you see the irrigation head hits this side of the tree, and the other side of the tree is dry. You're already starting to see the effects of drought stress on the tree. You have very little benefit from that soil injection of imidacloprid. And then in very rare cases on low infestation, we'll actually do a trunk injection. We've had parks that the infested tree's been in the middle of a playground, got wood chips. We're not going to introduce a trunk spray in that situation. Yeah, these are pretty uncommon. Moderate infestation, reproductive hosts, and Fusarium is active. We will always go with some type of bifenthrin, always bacillus sometimes tebuconazole, pentobark, and Nufilm. I will say in the last 2 years, I don't think we've used tebuconazole at all. The C seems to do a pretty good job. The ones that are highly infested or kind of moderately infested and moving to high infested, we have elected to trunk inject them. And because 2024 was such a terrible year for us, we did appreciably more trunk injections in this last year than we had done before. So severe or greater, I will say we've treated some severes and had some results, and we've also treated them and had no results. Again, the client determines the decision on how the treatments go. But again, the host condition, what the tree condition is, really impacts what treatment you're going to use. These trees are in terrible condition. You can almost expect that like a soil injection of imidacloprid is not going to be taken up. So it's not good money. So here's kind of the money of the deal. These are costs per DBH with no labor. So of course labor is going to factor into this, right? So this is just chemical. And I don't want to say that we focus primarily on chemical because we really don't. And in many of the cases, we go out with low infestations and they're clients that we go back year upon year upon year. We're monitoring those. We're not treating them. It's only when we get a moderate tree or an amplifier tree in there, we might choose to monitor every half year instead of going a full year. So I don't want to, hey, look at all these chemical costs. We're always doing chemical treatments. No, it's really not about the treatments. Our program is monitoring first. So going back to the cost, barrier sprays are about 29 cents an inch, soil injections 22, trunk injection with only imamectin benzoate, which we rarely do, is $6.15, a much more expensive product. And then the imamectin benzoate and propiconazole, $7.95. So you can see why we almost always put the propiconazole in if we're doing a trunk injection. It's usually a higher infestation, and the Fusarium disease is in the tree. So when you look at removing those trees, I don't want to spray anything. We have those clients that have strict IPM programs that do not allow any chemicals in their agencies, and they're removing the trees. So if you look at the cost of removing a tree, and then you go down to this 10-inch tree sample, and this is $30 per inch below 24-inch tree or $45 above. I just pulled that from one of our contracts. It's not to say that every contract costs that much. So that's just one that I decided to pull. But a 10-inch tree, if we trunk spray it and soil inject it, say it's a moderate infestation, that's about $5 per inch, okay? If we trunk inject that, that's $79 versus taking it out is $300. So of course the client is always like, well, you're coming back every year, right? You're gonna have to treat it every year, right? Well, right, but you're removing it. You're trying to preserve the aesthetic of the space. And having been the park superintendent in Long Beach before I had this job, I can tell you I removed over 1,000 30-inch plus sycamores that had been there for 80 years. It's a terrible situation. It really is a terrible situation. This was before some of these protocols were worked out. So there's a mix in here of treatment, non-treatment, monitoring for sure that allows us to be able to catch things when it is only a trunk spray maybe or a soil injection, when it's not very costly, it's a very small amount of chemical, and we can have a large effect on that. These are some of the sprays. I think they have ripped off our pictures. I think those are our applicators. So a little props for our guys. But these are the similar types of, and I have some other trees here I want to show you too. But I found this very interesting. I was going through some of the slides in preparation for the talk, and when I looked at this tree, and it has the same branch. We treated it in 2022 for a trunk spray and I think a soil injection, and we recommended that that branch be removed because it has ISHP. The trunk, you can see the hits are still, they're dry hits, so the tree's managed. It's got a nice canopy. it still has this branch, and it's still the same way. So I thought that was kind of interesting. So in the same park, same thing. We had done treatments in 2022, hadn't done any treatments, but only monitoring since then. There are some trees that are starting to show signs again, so we're probably going to start to make our list this fall. We're not going to treat now because the flight season's kind of getting to the end. We're going to go back in the spring and start to get these lists set up for treatments. So last time I gave a talk, I used this, and this is a park that we had not had on inventory. A call from a client, the trees look bad. I went out there, I'm like, wow, it's like an ISHB bonanza out here. So there's 271 trees, 30% had a low to moderate infestation. This is in 2024. And the amount of trees that had this were really stunning. I never seen it on the Palo Verde, and I've never seen it on Buckeye. So I have pictures. These are the pictures that I took in 2024. This is how we determine what treatments were going to be made to these trees. People are like, oh, that tree's in bad shape. But these defoliate when it's really hot. So my client's like, well, that thing's dead. You go out, well, let's see, I have this picture. I have a picture from the spring on this one, but if you go out there now, it's completely bare. Bad program didn't work. No, that tree is supposed to look like that right now. So we got this tree. It had ISHB really on all of these stems. We went back in 2024, we did a trunk injection. Again, I'll explain some of these things as we go through. In the spring, this thing recovered nicely. It kind of has a wonky canopy anyway, but we had good results on this tree. I went back, and this is just last week, and I saw that hit. So we will be monitoring this tree and trying to decide. Remember, on a trunk injection, you're going to get a couple of years out of those treatments. So the fact that this tree improved and that most of these stems don't have hits is a good success, but that's a little worrisome. So we'll be back to look at that tree to determine what we want to do next year. This is a Coast Live Oak, had several hits. This is last year. This is the tree now. We did a trunk spray, soil injection in 2024, the trunk injection in 2025. I elected to do a trunk injection this year on this tree because it had ISHB. It responded well to the trunk spray and the soil injection. But we were, I don't want to say late, this is some of what I'm going to talk about in a minute, but there's a lot of constraints on a pest control business, right? So we were not going to be able to get out there until June. So we elected to do the trunk injection to do a more thorough treatment of the Fusarium disease, and it worked well. I love this picture for a lot of reasons. I had a guy email me after I gave this talk last year and say, why aren't you just taking out all the box elders? Those things are just going to spread the disease everywhere. And I said, well, because we believe that we can try and see, we're going to treat the entire site that has infestation levels, and we'll see what happens. That's the same tree. This is now, and then this is in June when we went back to take a look at it. Full canopy, bark's healing. Yes, does it have hits? It does have hits. It's likely to need another treatment, but all but 3 box elder trees responded to the treatment. And many of them were trunk injected, but they did respond. This is sycamore, really affected. This is a year ago. This is the same tree. Yeah, you can see a lot of the hits still have live hits, a lot of dry hits on these trees, but in general, canopy is still very much intact. This is Palo Verde, like I'd never seen that. That was just weeping and bleeding out of a thinner canopy. That's a year ago. Same tree. This is a soil injecting it. It's kind of not a great structural tree. There's not a full canopy, but in general, it's a healthy tree. most of the hits on the bark have healed. So this was trunk spray soil injection 2 years. This is my favorite. I do have favorites. I go back every year. I'm like, oh, you look so good. We've treated you 4 years in a row. You're looking good. But this is me taking a picture of this. This is last July. This is March when we went back to do the evaluation. Canopy not looking great. This is what it looked like by the time we get out to treat it in June. So maybe everyone sitting out there would say, why? Because, you know, there's obviously active infestation. This is spreading to all the other trees. What's the hope? Well, this is September. And that's September with a fairly full canopy of leaves. And then this is the other side of the tree. So I'd say that's a pretty good return. We obviously trunk injected this. The other part about trunk injections is we do a lot of pre-sanitizing of the drills. There's a lot of drill bits with us. We post-treat the holes. So this is kind of a religion, you know, is working with our guys, making sure we're not drilling into infected wood. They're getting good solid wood. We're, you know, if you're using drill bits for 30, 40, 50 trees, you begin to drill into that, you're really sealing the wood inside of those holes. So you want to make sure you're using fresh drill bits when you're doing that as well. So we do a lot of sanitizing of the bits. Some of these trees really heal well. Sycamores are ones that do. We don't like to drill coast live oaks because they weep and don't heal over very well. But then we use our inventory system to document the different types of treatments that we're doing, so that as we go back and look at the trees, we have kind of a history on what's happened on the trees. So again, we treated 78 trees in 2024, 3 died. All the rest had a fairly good response. We treated 54 trees this year, and all of them have responded. So the benefit is doing nothing, you're likely to lose trees, but every year, especially, you're likely to treat less and less, and then be able to monitor them as we go through. So post-treatment activities, if I know we've treated things and I'm in that area, I'll always try and move through and take a nice walk and see how it's going. But we typically start at some points of the year, especially now. I was out with a client last week. We were looking at liquid ambers for an unrelated project. Across the street are 3 coast live oaks. These are street trees, all 3 infested with ISHB. So he's like, wait a minute, why a coast live oak? I'm like, yeah, they get it too. So we've asked him in the next months, do you want monitoring done on not only your park trees like we typically do, but also your street trees to see if we have an infestation level that's beginning to spread. So again, determining how to get back and when to get back, this site that I just showed, we go back pretty frequently because of how many trees were infested. If you have all low-level trees, not likely to have a need to return anytime soon. Okay, so the vastness of the Southern California host, there are a lot of trees that get this insect that the insect likes. The monitoring programs are really time consuming. So from an aspect, if we can, like up in the Ventura County site, we were actually using agricultural commissioners trapping data when we started doing that project to begin with, I think 2 or 3 years ago. But it does take a long time to get out there, look at the trees, get them on lists, and then decide what actually needs to happen to them. So having staff that do, and we have inventory specialists at West Coast Arborists, and we have the treatment folks. Sometimes we pull the inventory specialists over to do these assessments for us. So it allows us a little bit of flexibility there. Decision maker awareness, I think, is still an issue. That's why I love that map, is to be able to send that out to our clients. Like, you are in an area where ISHB is known. But I might be out looking for South American palm weevil, looking for gold spotted oak borer, Borers-Fusarium leaf beetle, and then I see a whole slew of trees over here that have ISHB, and bring that back to the decision maker. No, I want you just to look for that Borers-Fusarium beetle. You have ISHB. It's like, I'll get to that later. But there's really no getting to it later. So, continuing to bring awareness to our client base as to the scope of the pests that are out there for their inventories. And then employing treatments in a timely manner. As Bea showed, there's generally 2 flights per season, and they're not necessarily strictly Orange County is these months, Ventura County these months. They can really differ. So when to put the material out can be tricky. It also, in terms of where our issues lie, in terms of being a pest control business, there's 2 flights per season. We have many other pest pressures, and the season for the barrier treatment to work is about 2 months. So I go out in February and they're not flying till June. I haven't really done anything. So as a pest control business, we are constantly aware of where these trees are, what the level of infestation is. When we're trying to get the barrier treatments down, we may be going out to do barrier treatments first because they're faster and easier to get down, and going back and doing soil injections later because we can flex our staffing that way. So, and again, we have a lot of other pest pressures, so we're not just treating for ISHB. In fact, a lot of spring work that we're doing now is gold spotted oak borer and the Borers-Fusarium leaf beetle. South American palm weevil, if you haven't heard, is now up in Orange County. So we've done a lot of work on that pest down in San Diego, but we'll be busy with that as well. So again, other things that are important, if the trees aren't healthy, if the cultural practices are not good, it really impacts what we can do. So pruning, are they pruning out ISHB infested limbs? We can come behind and treat those pruning cuts to keep beetle from reinfesting. Are the trees mulched? Again, a healthier tree, more control of the water, the soil water influence with mulch. Supplemental irrigation, is it uniform? You have dry spots and wet spots around these trees. And then compaction, I think, is really one of those real limiting factors that we have very little control over. We're doing more and more air spading as a company, trying to improve some of the soil health around these trees. But again, soil injections with imidacloprid are really impacted by compacted soils. So we have to make that determination. And then the other challenges we look at is public agency folks have one budget. They grid trim, they remove, they plant, and they do treatments with that budget. So bringing an awareness of an issue is sometimes not enough if there's not enough budget to go around. So educating and trying to bring an awareness about how to use that budget successfully. Sometimes trunk sprays are all they can afford, and timed well, that allows them to keep some of the trees as healthy as they can. And then the government process, there's pesticide use is getting tougher and tougher. Also, the sustainability management plan, SMP, is that what it is for the state? No pesticides anymore by 2050. All of these constraints on us as a pest control business and me as a PCA make it more difficult to do these treatments. And the other thing, this monoculture, I have an accepted tree list. We can only plant these 10 trees, and Bea kind of talked about that, is getting away from, one accepted tree list, or at least amending an accepted tree list to allow for, to relieve that monoculture stand of trees, introduce new trees into it, and then maybe if a lot of sycamores or ISHB reproductive hosts are represented on there, we can add some new trees into that. So I'm going to finish with the regulatory considerations. This is really one of the main things that I do for West Coast arborists. The pollinator protocols, so sycamores and poplar and coast live oak don't really produce you know, a pollinating type of flower. So you don't have foraging bees on these trees, but we have magnolia trees that get ISHB and they do have flowers, and you cannot treat them if they have foraging bees. So in that case, you have to remove the flowers prior to the treatment. And we've actually done that, put that in our proposals. So the other reason for trunk injections can be if you have high, you know, moderate to high infested hosts that have flowers. Trunk injections is a way to go around that to provide protection for the tree and not have to worry about the pollinator. There's no going around those pollinator statements on these labels. That's a finable offense. And then the PPE considerations, I kind of think about, I'm always trying to make choices for the people that do the applications, our applicators, so that they don't have exposures to harmful chemicals, or we can minimize or mitigate those exposures. But really, bifenthrin mostly comes with a warning label, requires a 2-piece coverall and a mask when you're mixing and loading. So there's really no way to get around that. Cease biofungicide, oddly enough, that's a Bacillus subtilis, that's an organism. That also requires a respirator because of the microbial proteins in that product cause sensitization for repeated use, repeated exposures, not use. And then neonics by license-only applicators this year. Interesting to see what happened to neonics in this state in the coming years. They tried to get rid of them on landscape uses a year ago. That was vetoed. So my understanding is they're still trying to push that through. So again, sensitive environments, Bear kind of went through this. Trunk injections and soil injections are good. If you're near bodies of water or high density public spaces, trunk sprays are much more of a liability in that case. And then also groundwater surface water regulations are pretty strict in this state. I don't think a lot of people really understand that. When you look at groundwater surface water zones when we write the recommendations, and we also look at the Endangered Species Act. So I don't know how I am on time. Am I still good? Wrap it. Brandon's like, no. Okay, so I'll just show you how this applies going into the public land survey system. In looking at the endangered species, so you have to consider groundwater. You have to consider an endangered species when you're writing a written recommendation to protect any species that may be harmed in the areas that you're treating. So this is where we are right now. I entered that into here. You can see I got my coordinates, 7 south, 7 west. And when you enter these into the prescribed system, if you're sitting at a desktop, you can go to the California DPR site, or I'm going to be here later, I have it on my phone. If you're standing at the application site, you can hit it on your phone, and it'll pull up all of the species that have some type of regulation associated with them. Here's the coordinates that I entered, here's the species, and then this is for baseline of bifenthrin product, use limits, and here's the use limits. So occupied habitat for bumblebee. You got a bumblebee in the area, that's occupied. No can do on the baseline. So you're like, well, Kelly, that seems kind of ridiculous. It does seem kind of ridiculous, except I usually call the ag department and ask for their input on these sites that seem like it's not a straightforward situation. So runoff, occupied habitat, runoff, irrigation, and then spray drift. So if, a lot of times you pull these up and they'll say no use limits. So you really have no constraint on it. That's. Questions?
We have an online question, Kelly. It says, what tree pruning cuts to reduce infestation? Has it been determined pruning wounds are attractive to ISHB? Under the impression that pruning wounds were not attractive as they are for IPS.
Is it, or for what? What was the last part?
IPS beetles.
Oh, IPS. Well, it's an unprotected part of the tree. So if we have that tree that I showed you had one branch, we could coordinate the trunk spray after the pruning wound, and we can protect that pruning wound before the tree has a chance to heal that over. Nothing else? Everyone wants lunch. Did you guys all answer the questions right? Or should I do seemed like a pretty nice way to go, right? All right, thank you.
Our next speaker is Noah Roche. She is Senior Environmental Scientist with California State Parks, leading the Natural Resources Program across 40,000 plus acres of 25 park units in the Santa Monica Mountains in Los Angeles. She manages a team of permanent and seasonal staff while overseeing key initiatives such as wildlife resilience, wildfire resilience, cannabis watershed protection, facilities ignition prevention, forestry, and ongoing natural resource management. He's an experienced ecologist and natural resource leader with expertise in so many areas that I'm not even going to go into it because it will take us forever. I'm just going to turn it over to you, Noah.
Hello. Thank you for having me here today. I think we just had a couple of really great presentations about management protocols and what to do and how not to panic when you have an infestation. And this is the side of the story from the land manager who suddenly found an infestation and panicked. So this is Tapia Park. So Tapia Park is part of Malibu Creek State Park in the Santa Monica Mountains off of Las Virgenes. And it's a really nice picnic area full of coast live oaks. And then surrounding the coast live oak picnic area, there's a riparian area with tons of cottonwood and box elder and sycamore. So you can imagine how that went. Okay, so we first detected an infestation about 2 years ago in the fall of 2023. We were having some regularly scheduled contract arborists come through for a job walk to do some trimming, and all of a sudden the arborist and I looked over and both saw some very suspicious staining on some box elders right next to the entrance kiosk of the park. And we both immediately jumped out of the truck and ran up to it, and yeah, definitely looked very ISHB-y. But just to make sure, we called our friends at the LA County Ag. We called Rachel Burnap and said, please come and check our tree and see if we're seeing what we think we're seeing. And she came on over and she said, oh yeah, yep, you got ISHB. And we were like, oh no, what do we do? So we activated our wildfire and forestry resilience team, environmental scientists and 4 forestry aides to go out and inspect the larger area around Tapia Park. They used the iNaturalist, Ventura, and LA County Bad Beetle Survey for their inspections. We also called our neighbors. So there was the David Gonzalez training facility next door. There was the LA County Fire Forestry Unit across the street, and there was a Salvation Army camp over behind us. And so we called all of our neighbors to let them know that we have an infestation. And specifically, David Gonzalez invited us in to do an inspection on their property as well, as a neighborly. By the way, your tree's stuck too. So We identified over 150 trees that were infested with ISHB to some level or another. And so then we started, so we considered this an early detection rapid response plan. So we're like, okay, we've detected it, we figured out which trees have it, let's get them. All right, so this is 2 years ago, keep in mind. So we had to work California State Parks, right? So we have a process we have to go through. There's different agencies and permitting regulations that you have to go through in order to remove trees in the riparian area, with all of the different considerations of, you know, sensitive species and cultural sensitivity, and can we or can't we use various chemical or cultural mechanical treatments here. So our decision tree is we don't use pesticides and fungicides, insecticides on trees, due to, one, we're in a riparian area, there's a high water table. Number two, we're really concerned about off-target effects, not just pollinators, but any other insect species that might be affected by the insecticide treatment specifically. So kind of off the table for us right now, you know, maybe something to consider for the future as those treatments become more targeted. But right now, the off-target effects are just too wide. So came up with a plan, created a protocol of, okay, this is what we're gonna do. We have to put it through our own internal CEQA review, which is called the project evaluation form. We identified that we are gonna have to have a coastal development permit to be able to remove this many trees within the coastal zone, which the park is within the coastal zone. We needed funding. How are we going to pay for all this? And so we went to the Wildfire and Forestry Resilience Program, and we requested an allocation specifically to be able to handle this infestation, which, by the way, this was the first time that we identified ISHB on state park land in the Santa Monica Mountains. We've kind of been wary and waiting to see what was going to happen and kind of on the lookout for it, but this is the first time it hit us. And so far, the only time. Yes. And then we were like, okay, we've got a plan, we've got a protocol, we've inspected all of these trees. Who's going to come take them down for us? Well, you know, we've got taxpayer money, so we want to be really careful and prudent. So first of all, we approached various fire agencies, and we were like, you know what? I've got a perfect place for you guys to practice your falling. And so we invited CAL FIRE, we invited the LA County fire crews, we invited the MRCA to come and see our beautiful site for training purposes. And they all came through and they said, wow, you've got some complicated trees here. So each of those agencies were maybe only going to be able to take a small percentage, maybe like 10 to 15% of the trees that were there. Everything was all tangled up together. There were trees that were, you know, already in the process of dying. They could buckle or crumble on you in unexpected ways. There were trees that were over facilities. So it was not the best place for training opportunities. And some of the trees were kind of deep into the riparian area as well, which makes it difficult to haul things back out. Another layer of complexity is actually Tapia Park doesn't even really belong to us, it turns out. So it's, we've managed it for the last 30 years. We kind of act like it's ours. We do everything from cleaning it up to collecting fees, but it's actually owned by LA County Parks. I think we did a land swap for some beaches or something at some point. But anyways, it's in the plan for us to take over management at some point, but that hasn't happened yet and everyone kind of forgot about it. So all of a sudden, as we started getting the CDP, the coastal development permit, they reminded us, hey, you might want to check with the landowner. And we're like, oh yeah, that's not us. So we contacted LA County Parks. We're like, okay, we're gonna do you this amazing service and take out all of these infested trees for you. They're like, that's great. You need a right of entry permit. Please pay us a fee. So we did that. Meanwhile, it took a year. It took a year to figure out permitting. It took a year to figure out budgets. It took a year to figure out how much this was actually going to cost and who was going to do the removals and how we were going to set up the protocols to be able to take these trees down. So that took us right up to December of 2024. There were also some unexpected things that we found as we were inspecting the trees. So we heard that, you know, you're not going to find ISHB in anything smaller than 3 inches DBH. Not true, by the way. You know, you're probably not going to find severe infestations in coast live oaks. Also, not quite true. So, especially the box elders, you know, they would have multiple leaders, and they have all these sprouts and suckers sticking out every which way. Every single one of those, even the ones as big as my finger, were completely perforated with ISHB hits. There were younger coast live oaks that were growing up amongst them, you know, under 6 inches DBH. Those were all covered in ISHB as well. So it was pretty horrific, to say the least. So this is a map of our project area. This is Las Virgenes. So you got Malibu down to the south, and you got the valley up to the north, middle of the Santa Monica Mountains. And so this is kind of our picnic areas with coast live oaks. And these are the riparian zones that have all the ISHB in them. You can see here the red dots are the trees that we ended up removing, and the yellow dots are the trees that we're going to monitor and wait. Contracting. So we brought in one arborist to do an initial estimate. They came up with over $100,000. They were going to use all kinds of fancy equipment. They were going to bring in a cool little excavator that's only 2 feet wide and can, you know, get under the tree canopy and, you know, pluck things out and move things out. And they were going to have rental and a big tub grinder and everything really high tech. And then I called another tree company to come and look, and they bid us for about a quarter of the amount, and they were just going to climb the trees with ropes and cut them down and haul them out by hand. So you can guess which option we went for. A lot less impact too, right? Because all that equipment, even though it's light equipment and it's meant to have a lower impact, it still has more impact than hauling things out by hand, just bucking them up and shouldering them out. We also had to come up with a way to score the trees to decide which ones were coming out and which ones were not. We had a mix of different species, we had a mix of levels of infestation, we had a mix of where they were on the landscape in relation to high value facilities. So this is our scoring guide. Infestation level, tree species value, native or non-native, you know, box elder and willow versus oak and sycamore. Tree habitat value, a riparian area, oak woodland, or, you know, open landscape that's not really, you know, providing habitat. Tree health level is, aside from the infestation with ISHB, is it likely to come down on its own in the next 2 years or not, based on our standard, you know, California State Parks hazard tree assessment protocols, not taking targets into consideration, just assessing the tree, whether or not it's likely to fall in the next 2 years. Tree hazard level, whether there are targets or not. The host type, whether it's a reproductive host or not, whether it's likely to become an amplifier or not, whether, you know, based on the research that's come before us, whether it's likely to recover or not, right? Box elder is not likely to recover. Sycamore, quite likely to recover. So, yeah, basically we gave it a value. If it got 2 or more points, it's come down. If it got 1 or less points, then it stays. And if we're taking it down, we're removing it completely, we're chipping it to less than 1 inch, and we're solarizing those chips for 6 months, or at least that's what we thought. But it turns out nobody really has a good way to chip anything to under 1 inch. It doesn't exist as far as we've figured out. Every arborist we talk to, every fire agency we talk to, every chipper company we talk to, there's really no consistent path to getting those chips consistently under 1 inch, unless maybe you sift them and burn the rest. We had an air curtain burner that everybody was really excited about, but we couldn't get an AQMD permit to run it, so that was out the window. We were gonna just pile everything and solarize it and just take up our high value earning parking lot for the next year. Our visitor services didn't really like that idea, but they're gonna have to get on board. But, oh, and we called all of the composting facilities around the area to see if they would accept our chips. And they all said, no, sorry, you're a public agency, we're not gonna accept your chips. And also, they're all full of ISHB and we can't take them. So we were kind of stuck. Luckily, our contractor, BC Trees, that we ended up selecting, they said, you know what, we have a relationship with Agromen, one of the composting facilities in Ventura County, and they'll take them from us. So we had our contractor haul everything in a closed truck bed to Agromen, and that solved that problem. So end of December, we're ready to go. Our environmental scientist, Megan Edick, who spent the last year setting all this up, she's ready to go. Everyone's really excited. January 1st, we've been putting pressure. January 1st, we have to start these removals. We have to start these removals. December comes and the Franklin fire comes through. It actually was threatening to burn Tapia Park to the ground. It was getting really close. And kind of we were saying, you know, maybe it wouldn't be so bad if the ISHB just all burned down. But on the other side, we also have to protect our cultural resources, and we actually have a very important cultural facility in Tapia Park. It's actually like all of the museum collections that we have for the entire district. So we had to defend it. So we had to ask the fire agencies to defend that park, and it didn't actually come all the way over that way, but it got close. So we were all nervous and excited at the same time to see what was going to happen. My staff and I were actually fire line resource advisors. So anytime that there's an incident on state park land, we deploy as advisors to the fire agencies, and we advise them on where the sensitive natural species are, as well as cultural sites are, so that we can avoid them in suppression. And then we also work with the fire departments to do suppression repair and repair all those dozer lines. So we were pretty busy. But then that was over, and it was Christmas, and we were like, okay, let's get to work. Oh, there was another fire. It was the Palisades fire, and this time we were real involved. Our parks burned, I think, over 8,000 acres. And we lost like very important cultural facilities at Will Rogers. Not only me and my staff, but 60 other resource advisors from around the state park system came through, and yeah, we had our hands full. And Megan, she was like shaking with anger because she'd spent all this time prepping this project, and we really wanted it to get done before the nesting bird season comes in March, and it just didn't look like it was going to happen. We started begging every fire agency, like, if you have spare time and you want to do some fallen, we've got an opportunity here for you. If you see a tree that's marked with blue, go ahead. So we did actually manage to get some trees down that way. We also had some of our resource advisors do some of their practice falling. We had some tribal monitors from the Chumash tribe, their fire department. They came through, they did some felling for us. And so we got some trees down. Not all of them, not the majority of them, but we did get some trees down during the fire response. So finally, the fire was over. Megan decided to leave the department because the pace of being able to do something in a public agency was just not what she was expecting, right? So she was gone. I had a team of mostly seasonals in the Woofer program, and so I turned to my seasonals. I'm like, you know, you guys gotta grab this ball and run with it, and they did. They picked up the ball and they kept it going. They got the MRCA to come down and remove whatever trees they could remove as part of a training program. I think they got an additional 20 trees removed. We got an interagency agreement with the Resource Conservation District so that we could pay them to pay our contractors, so we wouldn't have to go out to bid and spend a lot of time doing that. We got the contractor, BC Trees, ready to go. They were able to take down all the trees right before the summer of this year, chip them all up, take them all over to Agrimen. Job done. Our staff did all of it was now in height of nesting bird season, so our staff did all of the nesting bird surveys in the dense riparian area. And the site is ready for the next phase. We also had to do some public outreach because we were going to do all these tree removals. We and LA County Parks were concerned that people were going to freak out that we were taking out over 70 trees. So we came up with some literature and some social media posts and a QR code that we posted all around the park and also on our social media feeds. We got some volunteers to come in and help us do some of the surveying and assessments. Some of our state park docents also participated. We learned some valuable lessons. So there's a little story here. So, okay, when we do our hazard tree assessments at the state park, we use numbers. So we use a big blue painted number on the tree trunks to identify which trees are coming down, right? So when we started marking the trees that were coming down for this project, we were like, okay, we shouldn't use numbers because we use numbers for our hazard tree surveys. Let's use letters. So we started marking letters. We went A, B, C. We got to Z, and there was still tons and tons of trees that were gonna fit our protocol for over value 2 to be taken down. So we had to go A1, A2, A3, all through the alphabet. We learned another valuable lesson. So we started realizing that a lot of these tree species are multi-trunk species, and not each trunk is affected. So some trunks are infested, some trunks are not, and we really needed to be able to mark that anybody can go out there and know which trunks and which pieces of material are coming down and being taken out. So we started putting the paint not just on the main trunk, but also on, you know, the additional multi-trunks, right? So we got to the front of the park and we were on K, and we came up to this beautiful multi-trunk, 3-trunked box elder. And so we marked each of the trunks with a K, and then we stood back at the front of the park and we're horrified at ourselves. So we had to turn back around, and then we just put Ks all over the tree. So then there were like 20 Ks all over it. It was fine, but something to be careful of. There was also some citizens that saw us kind of in plain clothes running around with spray cans marking trees and thought that we were a bunch of ne'er-do-wells and called the rangers on us, and the rangers pulled up and made fun of us. So that was fun. You know, early detection is key. When you think of your early detection rapid response plan, put in some patience, put in an extra P, because things might not happen as quickly as you want them to, despite your best efforts. The biology on the ground might not look like it does in the book. There's going to be things that don't fit what you were taught. There's going to be 1-inch diameter twigs that are full of ISHB that still have to be removed. Clarify land ownership before you start. You might think it's your property, but it might actually not be. And you might have to get a right of entry permit to do the work. A Our number one barrier was disposal. It was not possible to find somebody to chip under an inch. Solarizing was, the amount of material that we would have to solarize on the site would take up like a good baseball field of land. And that's either in a habitat area or on a paid parking lot. So not the best. Fire complicates everything. In state parks, there's always something coming up. Either there's a fire, or there's a flood, or there's a wind, or someone's freaking out about COVID or something. So plan for interruptions in your work. And seasonal staff really was able to carry this project forward and make it happen. And so don't discount or discredit the junior members of your team. They can take on a lot. Looking forward, the RCD, part of their contract is going to be monitoring for the next several years to see how this infestation is developing, whether we need repeat treatments, and whether the infestation will be reduced or whether it's going to spread. We also have to check around the edges with our sticky traps to see if the infestation is spreading outside of the bounds of where we know the infestation to be. Continued public outreach, especially in our firewood program. We've been working closely with our docents to create a program to have people buy firewood from our docents rather than from outside, and also working with our docents for where they're buying their firewood from, because we're providing firewood from Ojai, and it turned out to be a facility that wasn't having good protocols. So we had to contact that facility and make sure that they were checking their wood before they sold it. Restoration. What are we going to do and when are we going to do it? A lot of these are suckering species. So we cut them down. We didn't do grinding. We didn't do full removal of the stumps. So a lot of them are already sprouting back up. So that's part of our restoration right there. And then we'll wait a couple of years to see what happens, and then we'll probably plant a bunch of natives after the main infestation has died down. I think this project has really taught us a lot about resilience, perseverance, and persistence. And doing research and talking to your friends, and the UCIPM and the Bad Beetle Project, and all of our friends at the various fire agencies and our friends at the MRCA, we really had to reach out far and wide to get support for this project, both, not just like financial support, but brain support and knowledge support. So, yeah. Any questions?
Our next two speakers are Dr. Akif Eskelen. He's a plant pathologist from cooperative extension specialist. You're not a lecturer, you're a professor now at the Department of Plant Pathology at the University of California, Davis. I have this outdated. His primary research interests include the detection, biology, epidemiology, and control of fungal pathogens on the grapevine, fruit trees, and small fruits. And he's been involved in the shothole scene since the beginning. He's the one who I did polyphagus shothole for, this is something else, guys. And he developed a lot of the management practices that we've been talking about. So welcome, Akif. And then I guess are you guys sharing, or are you just going to be one after the other?
I'm just going to make it as short as possible, and then Paul is going to follow me, I guess.
Okay, I'll introduce Paul and do
sure. Okay.
Thank you.
So we didn't know which one is going to be the first talk, but I'm just going to make it as short as possible. My talk.
You might want to start here, and then you could use I know it just needs a little love here first.
Okay. So I'm not going to go through all these. This is basically previous speakers talk about all the options for the management of this pest and disease complex. Cultural sanitation, which is very important, that you all learn about it. Chemicals, there are a lot of options to manage, but there are downsides of it. They are not sustainable, short-term, and very expensive. We all learn about it. Attract and kill and deterrence. We have done a lot of studies, and other researchers in the other part of the world has done a lot of research. Actually, there's a very good study that has been done in Israel in avocado orchard. They try to attract and then kill the pest in that area, which was partly successful, but not 100% successful. You have heard about all these options. Now, I would like to talk a little bit part of the biocontrol. Why biocontrol is important? Is it sustainable? And it's going to provide some kind of the options for us. Paul, after me, is going to talk about the natural enemies. And I'm just going to focus on the use of entomopathogenic fungi, and then talk about the beneficial, naturally occurring endophytes within the plants, if we can utilize those beneficials to manage these pests. In the past, we have done a lot of studies about how we can control these beetles, including entomopathogenic fungi, which are the fungi that would attack the beetle and then manage it. Many of those entomopathogenic fungi are native to tropical climate. When you bring them into California condition, they are not able to function very well and then manage the control of the beetle. We even tried some of the foam, which is also to tend to keep the moisture and then food on the surface of the plant, where we can continue colonizing those entomopathogenic fungi, but they didn't work either. Those are like some of the adventurous management studies that we have done in the past. Defense activators, those are the ones that we haven't tried it. We did initial studies in the past, but Shannon and I are developing a project right now whether we can apply these defense activators to increase the defense mechanism of the host to manage those the pathogens that they are colonizing the plant. So very briefly, I'm going to talk about the endophytes. Endophytes are the naturally occurring beneficial microorganisms within the plant, like our gut bacteria. How our gut bacteria help us increasing the defense mechanism and also helping the digestion kind of thing. In the plant, they have the similar beneficial microorganism that help plant to defend against the pathogens. In the past, we have identified some escape trees in the parks where you can see the heavily infested trees next to the less infested trees. Shannon ran most of this project when she was doing her PhD. We collected samples from healthy and infested plant materials, looking into their the xylem inhabiting beneficial microorganisms from willow, cottonwood, oak, the sycamore, and all of those. And we're able to identify number of the beneficial microorganisms, which are mostly bacteria, to see if they are able to inhibit the growth of the harmful targeted fungi, in this case, Fusarium, Graphium, and Paracramonium. This is the control, this is the treated, these are the beneficial microorganisms that we isolated. When we plant them on the media, whatever the secondary metabolite that these bacteria produce, inhibiting the growth of the fungi in the plant. So when we first identified that, that was very exciting results, that we thought that, what if we can identify them and ferment them and then put them back into the plant to increase the defense mechanism. Because wherever these beneficial microorganisms are growing, the pathogen is not going to be able to grow. Why are we targeting the pathogen? Because those are the pathogens on the plants. They are the symbiotic pathogens, the fungi of the beetle, which are depending on as a food source. Without them, they are not going to be able to produce to their offsprings that Shannon and Bea talk about them as well. We also try to find out if those beneficiaries are going to target off-target microorganisms, like the Borers-Fusarium canker pathogen, which is the very common canker pathogen. They are also inhibiting those beneficiaries, the harmful fungi. So these are the common beneficial microorganisms that we isolated within Bacillus, one of the one that is the most common. Keep in your mind that the seeds, which is commercial version of it, which is the Bacillus species, which is different one. But these are different Bacillus which was identified from within the tissue of the plant. We injected or delivered those beneficials in the avocado seedlings in the past to see if they are going to be able to establish and then protect those plants. They were able to colonize in the healthy tissues. When we take them back, put them in a plate to see if they are still alive, they were able to colonize and then concentrate it and then inhibit the pathogen in the plant. So in this framework, it's working in the place. The question was, how are we going to increase the amount of those beneficiaries? So we have been working with a group of the researchers from South Korea, where they have identified how to ferment those beneficial microorganisms in a site open area by just increasing the certain nutrition into the medium. We were able to do that as well. And there's another way also doing it. It's a commercial way, in a sterile way, but this is the one that we have been trying as well. Another question is that how are we going to apply these beneficial microorganisms in the plant? We are thinking about the two ways. One of them, for the new planting, majority of the native plants in the native plant nurseries are being produced by the cuttings. We tried to deliver those beneficials into cuttings under the pressure, negative pressure, to get those beneficials diffused into those cuttings, where we can plant those into the out with a lot of kind of vaccination, beneficial microorganisms within that. We have started this project in the past, but we ran out of the funding, and then now there's another opportunity that Shannon and Bea has been working on, that we might be able to get some funding and then continue working on this part of this project as well. Another question is that that's okay, we can apply those like the new planting. How can we apply on the growing out in the plant? We are thinking about the two ways of it. One, we can apply as a soil injection. We know that some of those beneficial microorganisms could be also present within the rhizosphere, where they can produce secondary metabolite that could be picked up by the roots and then provide the protection on the plants, or injection in the plant in what time. So what is the difference between injecting the beneficial microorganism versus chemicals? In chemicals, you have to inject many times within the year or every year, because they are very short solution, and then chemical is going to diffuse and then disappear within the plant over the time. Versus these beneficials could colonize and then stay in the plant and then protect the plant in a long time, sustainable way. That's like the differences between beneficials and then also application of the chemicals. I think that's all I have to say. And I'm just going to give the stage to Paul to finish it so we can have our lunch. Any question for you? I'll say
Very, very excited about it. All right. So now, just a round of applause for each of the continued events. Paul Rodman Jones. He's a project scientist in the Department of Entomology at the University of California, Riverside, with extensive experience in DNA-based identification and population genetics with a broader context of invasive species management and biological control. He's currently involved in collaborative projects investigating the potential for classical biological control of invasive shothole borers in Diamondback moth in California. So, Dr. Rodman, please.
Hello, everyone. Thank you for coming today, and thank you to Randall for giving me this opportunity to give you an update on our work at developing classical biological control program for these invasive shothole borers. As you can see on the right there, that's a teaser for you. So for anyone in the room who doesn't know what classical biological control is, I'll start with just background on that. So effectively in classical biological control, it seeks to identify in the native range of the pest natural enemies that actually keep that pest in check in its native range. And if you can then identify those natural enemies, we then seek to bring those natural enemies to the invaded range, to here to California, with the hope that it will bring about similar control of that insect, in this case the invasive shothole borers here in California. So we identify the pest, we identify its native range, and we go to that native range and we start looking for natural enemies. We find natural enemies. There's a process then where we bring those natural enemies here to the US under permit from the USDA. And once we have them here, we can start to characterize those natural enemies. The first and probably the most important part of that is, can we rear those things here in captivity? Because if we can't rear them, we can't do an assessment of them. We're not going to be able to bring them in place. If we pass that hurdle, we then are in a position where we have to start conducting a series of non-target testing. We want to make sure that we're not going to release anything here in California that might be harmful to California native species. Best examples of that are all down in Australia, where my countrymen introduced a whole bunch of rabbits, cane toads, whatever you want, take your pick. So we want to make sure that what we are releasing are going to be safe. If our potential biological control agents pass those sort of tests, the next stage is we apply, we petition the USDA for a permit to release those. That triggers an environmental assessment. They want to know whether, if we're going to do this, the potential benefits outweigh the risks. And then, assuming we do get the go-ahead to release, then that necessitates that we implement monitoring to make sure that what we're releasing is establishing, and if it's establishing, isn't having any effect. So those are a simple sort of the route map, if you like, from start to finish of implementing and developing this kind of classical biological control work. Okay, so I want to now tell you a bit about where we are with this work for ISHB. So we know that ISHB is a complex and cryptic species. There are two here in California, Borers-Fusarium shothole borer, Borers-Fusarium shothole borer. There is another one, T shothole borer, which is invaded Florida and Hawaii. All of those are native to Southeast Asia. It's quite a big native range. And we've been conducting foreign exploration now for almost a decade. So far, we've visited 4 different places. We started in Vietnam 2014, accompanied by Keith. But I'm going to tell you today about the 2. They're slightly out of line, but the two destinations that we've had the most success. That's Taiwan and Thailand. I'll start with our work in Taiwan. So this is work that was originally proposed by Richard Stouthammer, my former PI. It's work that was in conjunction with a professor at National Chung Sing University in Taichung, Susan Tuan. And so they went to Taiwan. Which one's the point? Which is the point? So they went and they identified areas around Tainan City here, which is an area where they grow a lot of avocado. They identified ISHP infested areas in that region, and that kicked into then a process of visiting those areas, collecting infested wood, packing that wood up, shipping it under permit to a quarantine facility in the US. That work involves a whole team of people on the ground in Taiwan. We can't simply get off a plane in Taiwan and start cutting people's avocado trees down. I don't really like that. And so it's all the sort of collaborative element of this is sometimes understated, but simply without the likes of Chloe and Yi-Chan, native Taiwanese speakers, we couldn't do the work there. We can't go to a farmer and ask him in English if he minds if we sample his orchard. So these people are really the drivers of the work in Taiwan. And this, if anyone knows Ricky Lara from the CDFE, worked on this project for 1 year. We spent a year in Taiwan. So they collect this material. This material is then shipped to a quarantine facility in the US. Initially, that was being shipped to the quarantine facility at UCR, and at the time, that was part of Dina Hussain's PhD project. When Dina graduated, we actually moved the destination of that wood, so it was no longer going to UCR. It was actually going to the USDA quarantine facility on the East Coast in Buzzards Bay. And there it was looked after by a former PhD student. I was a time postdoc of mine, and now an actual employee of the USDA, who is effectively my boss now, in terms of she controls the ISHP grant program at the USDA at the federal level. Okay, so when that material arrives in the US, we take that material, we isolate the individual logs into these emergence jars, fancy jars, and then those jars are checked on a daily basis for the emergence of any natural enemies. So we get beetles coming out of there, we identify beetles, and anything else that we think fits the bill as being a natural enemy, we also collect. And then that material, those potential natural enemies, we introduce that into another emergence jar, and in that emergence jar, we put in there a bolt of wood that we ourselves have infested with only ISHP. So the material coming from the field, okay, we might get parasitoids, we might get natural enemies, but we have no we can't necessarily make the connection that whatever comes out there has parasitized or preyed upon shothole borer. But in that that non-target next test, where we put it only on ISHP infested wood, if we then get second generation, we know that it must be using ISHP as a host. And so through that process of non-target testing. We found 3 common parasitoid species, all of them new to science. We had a braconid, we had a bathylid. Both of these have proved incredibly difficult to rear in the lab for different reasons, which I won't go into. Feel free to ask me if you want to know. But the one that we're most excited about is the one that we've named, Phimasticus nudus draco, the naked dragon. So there she is. And funnily enough, this was in terms of emergence from the material that we brought back from the field. She was actually the least abundant. But now that we've learned a little bit more about the biology of this wasp, we actually see very good evidence in the field of parasitism by this wasp. And this is that evidence here. So what you have here is a beetle gallery. And this is the female's butt sticking out at that gallery. I think we saw earlier there showed a picture where sometimes you see the beetles protecting the entrance to the gallery. And you'll see that she has a hole in her butt there. That's the hole that wasp and its brother make when they exit out of the beetle. And so these wasps are endoparasitoids. What they do is they attack female beetles as they're starting to produce a gallery, and they'll lay typically they'll lay 2 eggs in a single beetle. One of those will be a female, one of those will be a male. And so we're not sure at this stage whether that brother sister, whether they mate inside of the beetle before they exit, or whether they mate as soon as they exit. We haven't actually witnessed the actual mating of the wasps. But good news, dead beetle. Dead beetle that's also just boring in to make a gallery, so there's no more beetles beyond because she hasn't produced a brood at that time. Okay, so moving on. In 2023, we extended the range of our survey to northern Thailand. So we've been working in this area around Chiang Mai and Mae Hon Son province. So mountainous region. They've planted a lot of avocado recently in this region. This is formerly a big opium growing region, and there's a whole bunch of government programs aimed at replacing opium farming with avocado farming. So again, we concentrate, when we went here, we concentrate on avocado. Again, right, driving around the streets in Southeast Asia, just like driving around the streets in California, you see castor bean everywhere. It stands out, you stop, you look. So we've been concentrating on avocado and castor bean mainly. And we're only really getting going with our work in Thailand. We haven't imported any material yet. I have now amended the permit, my import permit, to be able to bring material from Thailand. One thing that's missing in the equation from Thailand is there is no Kuroshio shothole borer there, or we haven't found it at least. But there is Prolificus shothole borer there, and there is T shothole borer there. And there is also, again, we got excited when we found this, Phimasticus nudus draco is also there in Thailand. And what I should have mentioned previously, so in Taiwan, I used DNA sequencing to identify that this Phimasticus was actually attacking both Prolificus and Kuroshio shothole borer. And it turns out in Thailand, we've been collecting it from wood that is only infested with T shothole borer, so it looks like it attacks the entire complex, which is good news for California, but it's also good news for Florida and potentially Hawaii. You'd think the federal government would be excited about that and give you some money, but they seem to be taking it away instead. Okay, so where are we currently at? Like I say, we've got this one wasp that we're very excited about. Here she is, parasitizing her, stinging this beetle with a bud, laying her 2 eggs. Phymasticus nudus radical. We have a colony now established in Buzzards Bay. We're about to restart importing material direct to UCR to establish a second colony there. Believe it or not, it's in terms of red tape, it's easier for me to import the material from Taiwan again, rather than try and get a shipment of our existing colony from Buzzards Bay to California. But that's also good, because it probably means that California will end up with a different genetic sampling of Phymasticus. So that's good. And this is really important because this picture here literally is our Phymasticus colony. This isn't something that we're going to be able to mass rear and release. We're going to rear it it'll be reared in small numbers and sort of trickled out, with the hope, should we get a release permit, with the hope that it will establish and multiply itself. And the other reason that this is important is back in February, lots of USDA staff were laid off temporarily in the end, but we actually nearly lost this colony due to a mite outbreak, an outbreak of mites in these jars. Thankfully, because we still have Yichang on the ground in Taiwan, and we know where to look, when to look. It was right at the end of the season for collecting Phymasticus. We went straight out and collected wood, and we got a whole bunch of them. We've restabilized that colony. We know that it attacks the whole complex, and right now, Chrissy across in Buzzards Bay is starting non-target testing. And that's another reason for re-establishing colony at UCR. So it's one thing to do non-target testing on ambrosia beetles on the East Coast, but of course we're not going to release it in Massachusetts. We're going to release it in California. So we want to make sure it doesn't attack anything here in California. And in some ways we're lucky, because there isn't really too much here that is closely related to the Ulysses. So we think we're going to be successful with that non-target testing. So again, just going back to that original slide, this is where we're at. We've identified and reported natural enemies. We know something about it. We're able to rear it, and we're now at this point of conducting host-specific testing. That itself is going to be a 2-3 year project. If we're successful with that, we'll seek approval, and hopefully we'll get eventually to the point where we might get a permit to release this. But in the meantime, we're not resting on our laurels. Again, this is just one species that we've got, and there's no given that it's gonna pass all the hurdles, and there's no given that if we release it here, it's going to establish. And so we're continuing our work in Thailand, but one further place that we really keen to work in is the Ryukyu Islands, and that's because we know that both Kuroshio and Polyphagus are present in Okinawa, at least, and we know that Polyphagus shothole borer there is a significant pest in mango. So we're currently looking for funding that would fund research to go there and see what we can find. And also closer to home, something that I do have funding for, and I'm going to start next month, possibly November, because I'm going on vacation next month, is looking for conducting a survey here in California in infested areas for natural enemies that might have switched and moved on to using Polyphagus or Kuroshio shothole borer as a host. And this comes out, there are lots of potential explanations, but when you see things like what we've heard about already in Tijuana River, taking out 150,000 trees, and then that area recovering, and now it's coming back. And that could be that could just be simple boom and bust sort of beetle population cycling. We don't know. But another alternative is that it might be a native natural enemy which is affecting some level of control and at least pushing that boom and bust cycle. And during COVID, we did actually collect some wood down in the Tijuana River Valley. And we did, at the time, we had one parasitoid emerge from that wood. Again, we have no basis to say that it definitely came from shothole borer. But we had a parasitoid emerge from that, which was a native parasitoid of a native beetle, the walnut twig beetle. It came out of willow. The walnut twig beetles named the walnut twig beetle for a reason. unlike the polyphagous shothole borer, it likes walnut and only walnut, or at least only evidence things in the juggle. So again, our aim is to survey areas in California, and if people are taking trees out and they have wood that they want to give me, infested wood, then I have a permit to take that to INQ, put it into quarantine, because we don't want to take stuff from San Diego County and introduce it to Riverside County if it's not present there. Not least Kuroshio shothole borer. Thankfully, we don't yet have that in Riverside County. So you can, if you have material that you want to remove and you think that it might be of interest to me, you can email me, and I will come collect that. Bear is also already helping towards that, folk at OC Parks. And yeah, I think that is it. A whole bunch of people that have been an important part of this work. Thank you. So, I mean, in terms of the non-target testing, we anticipate it's going to be at least 2 years to complete non-target testing. If that goes successfully, we put in our petition, and it would probably be another 2 years before we would get the go-ahead to release anything. I mean, even just so just as an example, so the permit that we had to import shothole borer infested material from Taiwan to Riverside, that actually expired when Richard retired, and I had to reapply for a new permit. That took 14 months to get. Even though it had been previously approved, it took 14 months to get that new permit re-approved. So things don't happen. None of this happens overnight. But, yeah.
Any other hope from vertebrate control?
From vertebrate control. I mean, it's not something we specifically looked at. I mean, I suspect that birds probably do feed. Certainly they would benefit from picking beetles off the surface because they're crawling around on the wood. I think the actual galleries themselves probably extend too far into the tree for your average bird to be able to reach. I mean, with bark beetles, where they can just below the surface of the bark, it's something different. But with this, it just goes up a little bit too far into the tree. I suspect lizards are probably a good option as well for picking beetles off the bark of trees.
Right. Well, thank you all for coming. For those who are online, and I apologize again for the hiccups we had up front. Hopefully, this was very useful for everybody, and we appreciate you attending. We appreciate all the presenters. Fantastic information, and look forward to learning more as these developments come about. And that concludes our meeting for today.